So we have about 2.5 hours. It's a lot of time. And I certainly have enough stuff to rattle on for 2.5 hours, but I don't want to do this really, right? And if we don't get through the program, that's fine. But what I definitely want to do at the end of the talk is come back to have a little bit of a discussion as to the degree what we have learned in the cholinergic system might apply to other ascending neuromodulator systems. And certainly, we know more, I think, now about acetylcholine and dopamine than we know about the other ascending systems. And so I think it's a good idea to end the talk, and I should not forget to do this, with a little bit of a discussion whether what we have learned here might be true for the other ascending systems. We just haven't really learned that yet. And why did we not learn this about the other ascending systems? Really largely, I think, because we do not have methods to listen real time to the release of the neuromodulator at the other ascending systems. Because I think the technology that allowed us to do that, both in the dopamine field and in the acetylcholine field, really has changed our views as to how these ascending systems work. So here is the program for the entire thing, if we manage to go through. And so let me just, a little bit pre-shadow the themes I would like to discuss with you. Number one, we will spend quite a bit of time talking about a fast version of cholinergic neurotransmission. And that fast version, as I will convince you, I hope, is not really neuromodulatory in nature, but it is actually a signal that is causing behavior. It is a deterministic signal. And I then will tell you that this deterministic signal works to a large degree by organizing oscillations, which can be behaviorally very, very powerful. And then we'll talk a lot about neuromodulation. And the trick here is to say it already and then repeat it a couple of times, that what cholinergic neuromodulation does, it does affect the probability and the amplitudes of these fast signals that cause behavior. Right? So in other words, cholinergic neuromodulation, through a step in between, as you will see, modulates fast cholinergic signaling, or this transience, as we call them, right? And once we have established the basics, then we'll talk about a bunch of translational fields. And we'll talk about the Coaly Transporter. The Coaly Transporter is interesting because the capacity of this transporter really controls the capacity for cholinergic neuromodulation, and that's why we are so interested in. And you'll see that we have been looking into various animals that have genetic variations of the Coaly Transporter. And so these animals therefore has various capacities for cholinergic neuromodulation, and that has direct effects on their ability, in our context at the very least, to control their attentional resources and processes, as you will see. Then we'll talk a little bit about Parkinson's disease, and specifically falls in Parkinson's disease, because those turn out to have something to do with levels of cholinergic neuromodulation, or better, with disappearing levels of cholinergic neuromodulation. And that will also bring us briefly to talking about cholinergic interneurons in the striatum. And lastly, we'll talk a little bit about an animal model of very poor cholinergic neuromodulation and therefore poor attentional control. And these are the so-called sign tracking animals, and that will bring us actually into the addiction field, as you will see. And the other thing I want to do is in the basic research part, in fact, pretty soon, I want to talk a little bit about the technology, right? Because we usually talk, so highfalutinly about data, but you often have no idea about how this data are actually being generated. And since we have remained the only lab, unfortunately, I have to say, that is measuring this fast cholinergic transience in performing animals, I really want to give you a sense how the technology looks like. And there's a similar technology, in fact, for glutamate, real-time sensing of glutamate release. The other thing I really want to do with this talk concerns the right column here. I want to try to illustrate to you how doing parallel and converging research in animals and humans really allows you to do some very important things. Number one, it allows you to double check some of the ideas you can deduce from animals as to the cognitive operations that are modulated by acetylcholine or that are being caused by this transience. Because if you only do animal research, right, you could get lost maybe in sort of rat or rodent cognitive neuroscience, and that would not be so smart, because in the end we want to translate. And these parallel research approaches allow us to cross-validate what we are doing, right? Very often the human research guides the animal research, but very often it's just also the other way around. And very often, and that's something we all have learned from Trevor, we are actually reverse engineering animal stuff and bring that to humans, and that enriches human research much better than anything else. That also applies to the Coaly transporters, because we have humans that have at least 2 Coaly transporter variants, and I'll show you that they come with distinct phenotypes that are perfectly consistent with the idea that low levels of cholinergic neuromodulations mediate low levels of potential control, and I will tell you what this is, while high levels actually will make you very strong at the attentional control side, even though there is, as always, a price to pay. And I cannot really not tell you about something we discovered very recently, which is that we can possibly assay the human capacity for cholinergic neuromodulation in human blood T lymphocytes, which is really, absolutely super cool. In Parkinson's, of course, we have our Parkinsonian fallers, as you will see, that are PET scanned, and that were at the starting point really to bring the entire cholinergic research field into Parkinson's disease. We have less to say yet about these sign trackers, this model for poor cholinergic attentional control in humans, but that's really in the works, right? So we got a lot to do, but I also got a lot of funny videos, right? So hopefully it will be okay. And if I see you, dozing off, we'll take a break anyway. So let's begin with the version, the traditional description of a neuromodulator system, just as Trevor introduced so formidably earlier this morning. And you know all these figures, right? But these figures have implications, right? So you have the cholinergic cell bodies in the basal forebrain area, and usually we have 1 or 2 arrows, right, to cortex and other frontal areas. And when you look at conventional depictions, for example, of cholinergic axons in the cortex, the whole system just looks like, it traditionally has been described, a monolithic, boring, diffusely organized system, right? That means, as always, you have a couple of thousand neurons in the basal forebrain, and they have the power to impact, all information processing in the cortex. But the idea is that that impact is the same all over, more or less, right? There's not much differentiation in what this couple of thousands of neurons tell the cortex. That's the traditional idea, right? And that's why we, for so long, have been stuck with functional descriptions that are, largely useless, maybe, like arousal, alertness, cortical gating of information. These are such broad definitions that it's very hard to really bring some science to those ideas. And in fact, Trevor wrote a paper once, which I always cite, and he doesn't cite anymore for himself, actually, which says that arousal is really a stupid idea, basically, right? Which I found a lovely argument. Now the anatomy is actually changing, right? It's just that the textbooks have not really incorporated this. And the person who has been spearheading this more than anybody else is Laszlo Zaborski at Rutgers University. So he has defined 35 morphologically defined cell clusters in the basal forebrain that all have their own projection systems. There is some topography, there is some overlap, but there's also a degree of specification we haven't really seen before. And that is really, I think, only the beginning of showing anatomical differentiation, because as you will see later on, the way we look at this system, and possibly not just at the cholinergic system, but at all of them, is that the evolving cortex has increasingly usurped or captured, if you like, the terminals of these ascending systems and incorporated those into local cortical circuitry. And so it's the cortex now that tells the cholinergic system what it wants to do with it, as opposed to largely, the soma and their afferent organization that tells the system what to do. And once you understand better, and we are still not very good at this, although I'll show you some data, how these synapses are really networked into the cortex, then you, of course, get a degree of selectivity and specificity functionally that is really unheard of. And Michael Hasselmo, of course, as you will see, has also spearheaded very much the idea that the best way to understand the function of the system is by looking at how they are being integrated into cortical circuitry. And again, to already say something about the end of the talk, I suspect the same is true for the brainstem ascending systems. We just don't know much about it. Okay, I want to start with one piece of old data because it starts really, it's a good beginning of the story I want to tell you. And it also allows me to introduce to you a task we need a lot in human studies and in rat and mouse studies, as you will see. And that's a task that is basically a signal detection task that has been designed to also give you an explicit false alarm rate. And over time, it's also a task that allows you to get measures of sustained attention if that's your interest. And so the way the task works is pretty straightforward. You have an intertrial interval, and then randomly there is either a signal or a cue or there's nothing. And cues vary by salience, right? Then 2 seconds, that depends on the species, of course, how that's being done precisely. But 2 seconds later, the animal is being asked, okay, what is it? Did you see a cue or perceive a cue or not? And it's being asked, in this case, by the levers coming out, right? And so the animal has always 2 answers. If there is a cue, it can say, oh yes, I got it, there was a cue, and we call this a hit, and it gets reward. Or it will miss it, right? And it gets not rewarded, and we get into the ITI, right? So learn this, please. It's important for later, right? If there is a Planck trial, of course, now it's the other way around. Now saying there was nothing, which is a correct rejection, that's right, and you get rewarded. And claiming a cue while there was none, that's a false alarm, not rewarded, right? And this goes on, let me just finish this, in humans, mice, rodents, for an hour, 40 minutes, whatever you want to do, a couple of 100 trials. Yes?
Is it always a visual cue?
These are visual, yes. You can also do it auditory, and people have done this. But with auditory cues, there's a bit more of an automatic processing of those cues, so it's harder to get decrements in performance over time. And the visual cue detection version is just more vulnerable for everything, right? Because the visual channel is just requires just more effortful processing generally than the auditory channel. So this is visual, right? So it's literally a light. I'll show you a video later on in a mouse. You'll see how it works, right? So this was a study we did a long time ago, 20 years ago, when we were able for the first time, as Trevor already mentioned, to selectively remove the cortical cholinergic input system, right? Which we were not able to do before. And the findings were kind of unexpectedly specific and spectacular. Because number one, the lesion had absolutely no effect on the animal's ability to give the right answer to blank trials, right? Correct rejection rate is absolutely unchanged. When you look at the hit rate as a function of signal salience, now after the lesion, the animal's hit rate is floored at about 30%. And I could explain those 30%. Maybe we come back to this a little bit later on, right? So they have a problem now saying, yes, I detected that signal, right? And we call this the detection of cues using a definition by Michael Posner, which is really a psychological definition, right? Detection means there is a cue, you incorporate it, and you respond to the cue, you report that it was present as you have learned before, right? That's the meaning of the detection of cues. This thing never recovers. We had animals for a year on this task. After the lesion, there's absolutely zero recovery of this, right? What's interesting about this is this giving you an idea, right? So whatever the cholinergic system is doing, at least in these kinds of contexts, it is something that has to do specifically with the detection of cues, right? While the default situation, if you like, there is nothing and you say there is nothing, that has nothing to do with the cholinergic system. The other important thing is that we found out back then that this is right hemispherically selective. If you remove the cholinergic system just from the right, you get the same result. You do it from the left, you get no such effect, right? Why is that? Well, we have known for a long time now that this kind of detection, sustained attention function is right hemispherically dominant, including in humans. And so once again, it's the cortex that tells the cholinergic system what to do, and therefore this is right hemispherically dominant. You had a question?
Yes, maybe it's a question. How did you remove the cholinergic input? Was it permanently removed?
Yes, this is a lesion with as Trevor mentioned, with saporin, which is a mitochondria incapacitating toxin. and that gets smuggled selectively into cholinergic neurons by binding to the P75 NGF receptor, which is selectively expressed by cholinergic neurons. This is something we have worked on with other people in the early 90s. It's a very well-established toxin you can buy commercially, right?
So what does it do? It just kills?
It kills the cholinergic cell bodies, yes, as Trevor showed before in the basal forebrain, exactly.
I have a question.
Yes.
So about the task, so they are able to see the cue visually, but they can't detect it as a significant, the behavioral significant stimulus? Like how?
So again,
So they are able to visualize a cue. They know it's there, but they don't identify as a behavioral significant, or?
it's behavioral significant because if they detect it, they get a reward. If they miss it, they do not, right?
But the animals that don't have the cholinergic input.
Oh, we get to that.
Okay.
We get to that. Of course, that's only the starting point, right? That's only the starting point, yes.
In this task, do the patterns of false alarms give you any extra information?
We'll get to that. All great questions, right? Super, absolutely super. We get to that. Okay, so it's right hemispherically dominant, and it's specific. That's the other thing, right? That is surprising when you think about this old piece of data. It's a trial-specific effect, right? A neuromodulator, you would think, okay, if a neuromodulator generally impairs attention in sort of a global way, you should see problems in both trial types, right? But it's specifically for these cute hit trials, right? So the neuromodulator does something quite specific, surprisingly enough, right? And so that's why this is a good starting point. So of course we were interested to see whether that's true in humans, but how do you test or find something like this in humans? And it took us only 20 years to show that this is also true in humans. And these data are from a group of patients we are now very interested in. We'll come back later on. These are Parkinsonian patients that on top of their dopamine losses also have cortical and thalamic cholinergic innervation losses. How do we know? We PET scan them using an acetylcholinesterase PET mapping approach, right? And so these subjects, they have, to the extent we can translate this data into losses, maybe 20, 30, 40% losses of their cholinergic system. They are not demented yet, right? Their MoCA scores are still fine. But when we now have the humans perform the same task, right, on a computer, it's the same thing except you're being prompted to give your answer with a tone, right? And we correlate this with residual innervation of the cortical cholinergic system. You see a very nice correlation, meaning the more you have lost in the cortex, the lower your performance is on that very task, the less you are capable of scoring these hits, right? So this is great, meaning that we can actually show loss of the system in humans does exactly the same thing. And this has nothing to do, by the way, with the losses in dopamine, right? As Trevor already foreshadowed also, and as we have shown, there's no regression on the dopamine losses. We'll come back to these guys. Yes?
So another question about the task. Do they have a time to press the level? It's like
yes, they have a limited time because otherwise it's an omission. But omissions are not being figured into performance measures here. The omissions are so low in humans. They are so motivated, right? They do misses, right? But they don't omit much, yeah. They have, I forgot, a second or so.
But so you measure the speed?
We do, but the speeds are not so interesting in that context, right? Okay? All right, so obviously when you have this kind of selectivity, something trial specific, you get to think, well, the cholinergic system must be doing something trial specific, right? But dialysis, and we'll come back to dialysis, which has its use here. Dialysis and the temporal resolution, you require to have enough of the analyte in your dialysate to actually detect it, just doesn't have the temporal resolution to really allow you to monitor the synapse, right? The release from the synapse. And so we worked for quite a while, and I will talk about this technically in a minute, on a method that will allow us to listen to the release of acetylcholine at real time, right? And I will just, again, say quickly the data we find, then we go a little bit into the methods in sort of a 5-minute excursus or so, right? And so this is exactly what we found, right? So here's a blank trial. The levers come out. The animal gives the right response and gets reward, but the cholinergic system is not interested, right? Just as you would predict from the absence of a lesion effect on this trial type. But if there is a cue and the levers come out, you can predict within a couple of 100 milliseconds precisely whether the animal will score a hit or a miss. Because if you now see a rise in the signal, you know that the animal will score a hit, right? Precisely so, right? And this is what we call this transient. And these transients, we come back to those in 5 or 6 minutes. They are really specific for this Q-hit trials, right? So they are the first piece of data that show that these transients do exist, which surprised us almost as much as anybody else. And what is definitely, really cute, given the old lesion data, is that they are specific for the trials. You would expect they are specific coming from the lesions. All right, so we come back to this, and I want to just step back, so you guys keep this in mind, and give you a little bit of a sense of the methodology, right? Because usually this flies just by. And maybe I can convince one or the other person to start working on this, because I don't want to be the only lab doing this for the rest of my career. It's just boring, right? So, yeah, we'll convert the masses, exactly, right? Right. Yes?
A kind of decision making is involved in this.
We come to this. We come to this. Let's do methods first. Let's do methods first.
In these experiments, have you blocked the dopaminergic system, the dopamine release, or just?
No. And I suspect it would have very little effect. But can we come back to all of this later, and let's do methods a little bit? Okay, so the idea how to do this actually has been around since a long time. People just couldn't figure out the technology for it, right? So the idea is that acetylcholine is coming out of the synapse. It's being hydrolyzed by acetylcholinesterase. And there is an assumption here we will discuss a little bit, meaning the acetylcholinesterase is definitely super potent, we know this, and it's also super abundant, right? So everything gets hydrolyzed, right? And so you're making choline, right? Extracellular space is already sitting in 3 micromolar of choline, but you're throwing a little bit more choline into that sea of choline, right? And that's what you can measure, right? And the idea is like so. You have an electrode onto which you immobilize choline oxidase, right? Which is a specific substrate to oxidize choline, okay? When you do this, a byproduct is peroxide, right? And the electrochemist is always happy when we can make peroxide, because we can apply a voltage on a platinum site and oxidize peroxide. And so per peroxide, we make 4 electrons. And amperometry basically means we are chasing these super small currents. And chasing these super small, picoamps, that's the challenge, really, right? Now, as I said, the idea was around for a long time, but the breakthrough really came from a group in Kentucky that built these ceramic backbone electrodes where you have these 4 platinum sites, right? And so you immobilize the enzyme onto at least 2 out of these 4 sites, and the other 4 you use for control, right? So you immobilize everything but the choline oxidase. And you see these things are pretty small, 300 by 15 micrometers. You insert those, right? And the idea is now you can specifically listen to acetylcholine release by measuring basically the product of choline oxidation, right? There is another protective layer that you have to put on these electrodes. And there are 2 of them, and I'll show you a slide there, because there's a big discussion in the field, what's better here, and it's a big deal which one you choose, because there's other stuff like dopamine ascorbic acid that's electroactive and can be oxidized and might give you a hydrogen peroxidase. So you have to control for this, right? So here's the scheme again. The selectivity comes again from choline oxidase. It comes from a layer like, in this case, MPT, amphenylenediamine, that protects or doesn't allow dopamine at all to go to the platinum site. You, of course, also calibrate this electrode in vitro, and I'll show you this, right? And then you apply a fixed potential to make sure that you are indeed measuring those 4 electrons coming from peroxide. And you can easily control this by just changing the voltage, because the voltage is specific for peroxide. You change it, you lose your signal, you're good, right? Right, so I just want to show you really practical, how we do this, right? So we have a station which you need to make your electrodes and wire them up, right? So all the undergrads learn how to solder and things like this and learn how to make electrodes. And of course, you need a standard lab with some special interfaces and regular electrophysics and so forth, right? The setup will cost you $25,000 or so, right? But it's, you know, it's good to know. But the running costs are high because these ceramic backbones are very expensive, and you have to buy them from a company in Kentucky, right? So here you see the electrodes. You can't see the recording sites because they are so small, right? But you see now the soldering and the wires attached to the 4 channels of the 4 platinum sites. And what we do, we buy actually electrodes that are made for use in anesthetized animals, but then we cut them off and put a head stage on top of it, as I will show you. And there are protocols out there for cleaning and doing all of this. So here you see an electrode. So here are the 4 sites, 1, 2, 3. And the 4th sites you can't see quite because there is a cannula coming down. We are using in vivo to pressure eject drugs to test the electrodes that they are still working, right? And the sensitivity is good, or sometimes just do neuropharmacology. And here you see an electrode. Here again is the electrode and the head stage. You know how it's being implanted. This is an electrode that got obviously ripped out of an animal, so you see how the whole things look like. And here again is the electrode, and here is a guide cannula, right? So that's how you do it. You implant it, and you're good to go. After some calibration, which I also explain in a second, right? So these barriers are important. MPD, or another one that's called Nafion, which is you know, heavily used in all forms of electrochemistry. And there are good and bad things about those, and the slides get uploaded, and you can study this if you want to. But one is better for long-term implantation, and the other one is better for acute studies, and both have their pros and cons, and you'll have to play with those things. But it's important to understand that these things have to be either baked onto the electrodes or using an electropolymerization step, basically glued on top of the enzyme layer. So in vitro, right, you show that the electrode doesn't respond to ascorbate and responds nicely to increasing concentrations of choline, because that's your reporter molecule, right? And it doesn't do much to dopamine, right? So this is obviously an MPD-coated electrode. You get a nice, good linearity, and there are certain standards you wanna meet for sensitivity and linearity and selectivity, right? But those data have been published, right? Yes?
Sorry, this might be a bit of a stupid question, but I don't understand how, if you're not applying, so you're only oxidizing, not reducing, right? So this is, you're applying...
You're only oxidizing, yes.
This is a continuous potential.
Yes.
But I don't understand how you get any temporal resolution, because from, you know, as in, so usually you oxidize the juice, and then at each time point, from what I know... Well,
you'll see if you have child-specific behavior, and that's actually a very important point, right? You always baseline your electrode prior to the next cue, for example.
Sure, of course,
right?
But then...
And the current will go away, right? You have your acetylcholine coming out, you have your handful of choline, the electrode now sees, right? So you get your spike, and then the electrode goes more or less back to baseline, right? So it's not like voltammetry in that respect, right? That's where you're coming from. That's where you're coming from, right?
There is no like additional stimulation. You do not stimulate.
No. I let the neurons talk. We just let the neurons talk.
And on that point then, so do you have to like cycle your electrodes beforehand? I mean, do you have to do the whole...
You can sample at 40 hertz, right? There is no limit, right? The only limit is your interface, right? So you can literally listen at real time, right? And it's much more straightforward in that way that voltammetry is, right? You see. Okay. Clever question. Okay. Does it really work? Yes?
How much is the resistance for this electrode?
Resistance.
In this kind of...
Oh man, I once knew this. I don't remember.
Sorry, just about the spike of...
We'll get to that.
The refractory period.
Excuse me?
The refractory period. I mean, spike, then refractory period, and then again
You can, okay, the way to test this is, for example, you can drive neurons with opto, right? And we have never managed to exhaust the electrode, never. Of course, at some point you see spikes building up, right? So it's not so interesting, right? But behaviorally, where you have a fairly low temporal resolution, right, because of the behavior, you're not running into any trouble. Wow, you guys are interested in methods. Who would have thought? Yes.
Can you do this in freely moving animals?
That's the only way it makes sense. I'll show you. Otherwise, I wouldn't be doing it, right? We are behavioral neuroscientists, not cellular mechanics, right? Foot drugs.
But it's not working like microdialysis panel. It doesn't make, you can't.
No. It's exactly right. I mean, you can also make, the technology also works for glutamate oxidase, right? But yes, that's the only thing you can do, unless you manage to put different enzymes onto different sites, which is very tough to do because it's a very small area you have, and you have to do it by hand, right? It's a real art, right? The best students in the lab are the best coders. Right, so those we like.
Once more.
Wow.
How deep in the brain you can put this?
Yes. So these ones are fairly short, but the Kentucky folks have now started to make long stem ceramics for primate research. They are hellishly expensive, but you can buy them, right? And the ceramic makes a difference because the backbone is stable, so this thing is not bending, right? And the way they fabricated these platinum sites, they're a little bit like bathtubs, so you get enough enzyme to become immobilized, right? Well, I thought I'd bore you to death with this. It's amazing. Okay, does it really work, right? Well, if you block the esterase by pressure ejecting a neostigmine, right, an esterase inhibitor, and then you measure potassium-evoked acetylcholine release, which gives you normally a very nice, of course, an easy spike. If you block the esterase from hydrolyzing acetylcholine, this thing is lost, right? Likewise, if you block the terminal from depolarization, right, neither potassium nor scopolamine, which is giving you a spike because it blocks the autoreceptor, right? If you block depolarization with TTX, you don't see it anymore, right? Or you can simply take out the cholinergic system again using the immunotoxin, and your potassium will not give you any spike, right? So you can do a lot to show that you are indeed measuring what you claim to measure, which is acetylcholine release, right? And we have done this. Which brings me to a topic that's not just technical, but now also conceptual, which is something Trevor already mentioned, right? Volume transmission has been considered a typical thing for neuromodulators, right? That's shown here. What's the idea? The idea for the cholinergic system is like this. Your acetylcholine gets released. These red little sharks is the esterase. But if you think that there is volume transmission, you have to assume that the esterase is not abundant enough to actually kill all the acetylcholine. So some of the acetylcholine will swim out and have distant effects, right? Outside the classic synapse. That's volume transmission, right? And there have been folks like Descaries who have proposed they are low millimolar. They would be huge, ambient levels or tones of cholinergic transmission, right? And I've seen in some of your abstracts, I think, the word cholinergic tone, right? So let's see what the story is about this. The alternative is that the esterase is so potent and so abundant that nothing escapes the synaptic cleft, right? And so we got interested in this and thought, how can we measure this? And we thought we can measure this by making electrodes that don't just have the choline oxidase immobilized, but also the acetylcholinesterase. Meaning if there is residual acetylcholine, right, we are now actually hydrolyzing this on the electrode, make locally choline, and the choline oxidase will show us that there is acetylcholine swimming around. And this was actually done by a student in my lab who is now in Cambridge in Trevor's place. Right? So here is one of those electrodes, right? Up here we code it just with choline oxidase, and down here we put both enzymes on it, right? And I'll show you in vitro this works, right? So if you just administer choline, right, of course both electrodes will give you the same response as both electrodes have the choline oxidase on it, right? But when you administer, still in vitro, acetylcholine, right, the choline oxidase coated electrode can do nothing with it, right, because there's no esterase in the bath. But the electrode that has both the esterase and the choline oxidase now sees it, right? Like this stuff was hydrolyzed by endogenous acetylcholine, right? So in the brain we go, And what we have done is we used potassium to make huge depolarization events, meaning we are throwing out all the acetylcholine we possibly can, right? I mean, it's a bizarre, powerful stimulus. But the idea is if there is acetylcholine that gets spared from the endogenous esterase, well, then our double-coated electrode will still see that, right? But the fact of the matter is, as you will see, that the double electrode in orange versus the single in red, they both have exactly the same responses, right? And so we cannot find residual acetylcholine, right? And so this data would suggest there's no volume transmission, right? But the stuff stays in a classical synapse, and the esterase will not allow volume transmission at all. You will see later on this cannot be the entire story, and it's a real problem in the field that we haven't really resolved, right? But it's a fair question to ask whether acetylcholine, in that respect, is a neuromodulator or not. And at least these data say, well, it's not, right? Because it's not volume transmitted at all, right? And there are lots of data in the literature that will disagree with that, by the way.
Lifetime of a given preparation.
Of a given preparation. Oh, this of course is in an anesthetized animal, right? This is in, sorry, I didn't catch this. This is an anesthetized animal. You can do this all afternoon, right? Basically, right? I mean, as long as the electrode is good. In vivo, in the behaving animal, it's a different question, and it's a bit of a different electrode. And there you have, after implantation, you wait 3-4 days until things are really good. And then you have a window of 5, 6, 7 days to do good recordings. Sorry that I didn't catch that question. Right? Thanks, Mike.
Is there a threshold for how much choline can be detected?
Is there a
threshold for the current detection of the choline?
Is there a threshold? That is exactly the key question, right? Because it could always, that's a real clever question, because it could always be that there's some acetylcholine that's escaping that's below a threshold for those double-coated electrodes. It's a very good question, and it's the key question. But it's definitely, our detection threshold is definitely below the millimolar area. So the Descartes proposal of this ambient level is definitely wrong, right? If there is something swimming out, we are talking very, very low single-digit micromolar and less, right, acetylcholine levels, right? It's a very good question. We have to move on. I never get rid of this talk if you get stuck with methods.
All the electrons shows that there is no any difference, so which means that there is no volume transmission.
Which means that at least up to our threshold, and she asked the key question, we cannot find it. There's always a threshold in every method, and you could always miss something underneath, right?
Whether it's related to the Acetylcholine Transporter activation or not.
Let's wait for that. Then we'll talk about the transporter later. Okay. All right. Back to this, right? So remember how the task works, and we are seeing this transience, right? So that's really cool and predicted.
Do you also see a trend in false alarm?
No. No. Okay. You guys are asking really good question. Here's the problem with false alarms, as you will see later on. False alarms rates are very low, 5%, 10%, right? Some of that is a component of that is random lever selection, right? That's underneath every level of performance. And the problem we have is I have no second way to say this was a true false alarm or just one of those random trials where the animal just checked out, right? Because sometimes we see a spike, right? That's why your question is so darn clever, right? None of my postdocs would be able to ask this question, right? But I cannot just then say, okay, this is a real false alarm, the rest is random, right? I have no independent way. Really good question.
If you have an increase in hits, do you have an increase in misses? And is it that the animal is disengaged from the task?
Can we wait? Many of these questions will get answered in some ways, I think. Okay, so number one, we were so happy when we saw this, except that we didn't see this in all signal hit trials, right? But in only like 70% or so, right? And so look at these signal hit trials. They're all the same behaviorally, right? But sometimes we saw it and sometimes we didn't. And for half a year, we were digging around, what's the difference between this trial and this trial and this trial? What, is the animal doing something different, right? Couldn't figure it out. until one day we figured it out, it has to do with the trial sequence. Oh my God, right? And what the story is that if you come off a hit, so you might have just had a transient, and the next one is also a signal hit, which we now call a consecutive hit, then you do not see the transient, right? You see the transient if your prior trial was either a blank with a correct rejection, or it was a cue that was missed, which in the animal's brain is the same thing, right? It's not there. If then you come, with a signal hit trial, which we now call an incongruent hit, then we see this thing, right? Why does that make sense, or would this be a good thing, right? So the idea is that during consecutive hits, the response is already online, the circuit is already set, just in those terms Trevor used. And you don't require a memory-based shift in response rule, right? So you're doing always the same thing, signal hit, you are there, you're doing it, your circuit is in the state. But if there's an incongruent hit, then you actually have to shift, maybe from even a default mode, which is blank trial performance, to say, okay, now there is a cue, and I've been trained, that cue requires a different response, right? So you're moving, as we say in psychology, from one of the terms we're using, from monitoring to cue-oriented responding, or from intrinsic to external attention, right? So it's a little bit like this, just to drive the point home, right? So you just have been traveling recently, right? You're waiting for your luggage. This is monitoring, right? This is perceptual, reflective, or internal attention. Your bag shows up, you match it with your memory, with a cue, and you basically go through the detection process, means, okay, this means I have to go there and pick this thing up, right? So this is not trivial, because if you are, in disease, if you don't make cholinergic transients, and therefore you cannot shift to this cue-directed behavior, you will be arrested in the monitoring mode, something you actually see very often in old people, right? They're arrested in the monitoring mode. They monitor, but they don't do anything with those cues anymore, right? It's quite incapacitating, really, right? We know quite a bit what's happening during those, not quite a bit, but a little bit, what's happening during these consecutive hits. And we think actually the transients are now actively suppressed. Why would this make sense? Well, if you make a transient, every time you do a cue hit, there's a risk that you're biasing the circuit towards doing detections, right? And that will reduce your flexibility to move between blank and cued trials. At least computationally, you can make that argument to a degree.
Sorry, can you tell the difference between there being no cue detection and no action upon cue detection?
It's part of it, right? I mean, that's the definition of cue detection, right? It involves the action, right? Meaning you allow the cue to direct your behavior in a way you have learned before.
I personally think there's a difference between sort of, I don't know, like actually...
I know what you're saying, and I don't disagree with you, but in this case, we have never parsed this apart, right? We built the action, as you will see, actually, we talk a little bit about this, into the entire process. We think it's mediated by this.
Partly related to this, does the Coaly transient arise during the cue, just before the action?
Well, no, you see this right here. It's a really good example, right? It's the cue that evokes it, right? And I figure I'm going to know what you're going to ask next, but we'll get to that question also, right? But it's precisely cue evoked, right? But again, if the transient fails to come up, the animal will miss, right? We'll get to that. I think I know where you're going with this. Okay, yes. So this is our second trip to humans, right? Because at that point, the idea that these cholinergic transients mediate these particular incongruent hits, that scared us a little, right? Because this is all rat work, and maybe it's just rats who have this thing about incongruent hits, but humans don't care about this stuff at all cognitively. So with my colleague, Cindy Lastic, we did an fMRI study where humans performed exactly the kind of task I described to you before. It's just a human version of it. And what I'm showing you here is the contrast between incongruent and consecutive hits. And what we found, just like in animals, is only right side activity, and this is in the right frontal cortex, Brodmann's area 10. And what we also found, and that surprised us, is actually we could even see activation in the basal forebrain as indicating this contrast, again, only on the right side, right? So the cholinergic system really likes in humans, just in animals, specifically these incongruent hits, and seems to be mediating specifically these incongruent hits. There was also functional connectivity with parietal cortex, which is nice to see, because that's the human, large attention system, is the frontoparietal network. And BA10 activation also correlated with faster incongruent hits, which is, as the fMRI folks know, a good thing, because it gives you some confidence that this really is not just an epiphenomenon, but really an activation that has to do with task performance, right? So we think these incongruent hits are actually significant from a cognitive perspective. And to the extent we allow ourselves to interpret this data in terms of cholinergic activity, given the basal forebrain activation, we would like to think that much of what we are seeing here in Brodmann area 10 is cholinergically mediated. All right, but these are all correlational data, right? And we would like to know, are these transients really functional? And so, of course, we use optogenetics to make them, right? But we have some advantages here as opposed to many experiments in the field. We know exactly how those endogenous transients look like, right? The amplitude, the rise time here in red. So we have a pretty good idea of what we want to make, right? Which is not the case in many opto studies. And so in gray, you see a couple of optogenetically evoked transients using various levels of intensity. And you see we can come pretty close to the red one, right? It will never look exactly the same, because after all, optogenetic stimulation is a very bizarre, unnatural thing, right? Because the specificity, while we always emphasize this, is also the craziness of the method, right? Because in reality, it's never the case that it's just the cholinergic neurons that are being activated. It's entire networks, right? And so it's logical that opto, when it works, it's amazing enough that it works given how bizarre it is, but at least we know exactly what kind of transients we would like to make, right? And we did this back then in cat-cree mice that, of course, got transfected with general-rhodopsin using the usual methods. So I show you a movie now with a mouse performing the task I described before. There's nothing happening here. I just want you to, again, understand how the task works, right? And you will actually be amazed how disciplined this mouse is. Here's a cue. He goes over. This was the hit, not a lever now, but it's this moving nose pokers, because mice like this nose pokers better, right? And you see this mouse is pretty focused to the intelligence panel, right? It's not like your usual crazy mouse going around. This was a blank. So he goes over, nose pokes the other side, which is the correct ejection, gets water again, right? So they are, once you get the machinery right and the parameters right, the performance is at the level of rats, right? Amazingly enough. They are just as smart as rats. We just have a harder time to understand how they tick sometimes.
Is there anything that tells them that the next trial has started? Or is it just
No,
a continuation of
no, the intertrial interval is variable, right? And so again, the command to tell me is when those two nose ports, the response devices come out, right? And so again, if there was nothing and the devices come out, well, then you go over to the right and make a correct ejection, right? But I want you to understand, again, if there is no cue and you go to the left, that's a false alarm, that's wrong, because that matters in a second, okay? So this is a pretty complicated experimentation, because animals have to learn the task, takes a long time, right? You put the vector into the basal forebrain, and you put the lasers either into the basal forebrain, or we also did it just into the right prefrontal cortex. And then they need to learn the task again while being tethered, and then you can do the task, right? It's nothing for students who don't want to take at least 7 or 8 years to get their PhDs, unfortunately. It's very, very tough stuff, right? So the first experiment we did is pretty straightforward. We made transients whenever there was a cue. Right, we know that the animals will make endogenous transients, right, if it scores a hit. But we just didn't care. We just made a transient in blocks of trials every time there is a cue, right? And what we see compared to non-stimulation, so this is averaged overall signal durations. This is non-stimulation levels. In a stimulation intensity-dependent manner, we increase the hit rates, right? So the animal makes more hits, which is really cool. It's even cooler when you look at the hit rate as a function of cue salience, because the shortest cues are only hit at about 50 or so %, right? And when you now turn the laser on, coinciding with the cue, make this transient, now you see all the benefit comes actually from an increased hit rates to these short signals, right? So that's cool. That's what you would expect if you really believe that these transients are functional.
How long was your stimulation?
The stimulation was exactly as long to make transients that looked like this, right? Whatever the parameters were, I would have to look this up, I think. Okay?
Isn't it variable? Duration, a stimulus duration?
It's variable. It's variable, yes. That's how you get at baseline this duration-dependent hit rate, right? And by the way, I want to say, very often in the literature, people say that they are taxing attention by varying stimulus duration. That's not really true, right? What you're really doing is that you are avoiding that the animal establish fixed detection thresholds, which makes the task more dynamic and harder. But you shouldn't talk, in simple terms of, shorter, lower detection rate means poorer attention. It's more complicated than that.
Did they get rewarded for correct judging?
Yes, absolutely, absolutely. Which is important because the reward is not contributing to making transients, and there's more data to that. Yes, a very good question. Yes, you are rewarded in a very symmetric fashion in that respect, right? And correct rejections never make these transients. So this is cool, right? It fits the idea. But I always thought the real hard test that these transients are functional comes from blank trials, right? If you have a blank trial and you now make a transient where normally never there is one, right? Just coming back to your question. And if that transient really forces this cue detection operation, once again, the big, large definition, right? Then you should see false alarms, right? Claims for cues while there was none, right? So normally, as I mentioned, false alarm rates are very low, even in mice, 15%, right? Bilateral stimulation of cholinergic neurons, so we're making bilateral transients. We are nearly doubling the false alarm rate as a function of stimulation intensity, right? But what's really cooler, I think, is you can just make transients in the right prefrontal cortex, right? We've seen before, this is a right hemispheric thing. It's largely prefrontal, we think, right? Look, just one side, we're making transients, and we make a lot of false alarms, right? This is really, for me, the proof that these transients are functional.
Is that prelimbic?
That's prelimbic. That's prelimbic. And I want to show you a video that is perhaps the most impressive video I have. There are funnier ones later, but this one is impressive. So what you see here is a bunch of blank trials, right? And for you to see the laser coming on, we didn't paint the back side of the head stage. So you can see the laser coming on. And the laser comes on just, just a little before the two response ports come out, right? And you will see that the laser drives the animals to the left side to score a false alarm. And of course, before you asked it, we counterbalanced this. Some animals did left, some animals did right, all of that, right? But it's really impressive to see, right? So here's the guy. Watch for the things coming out, but first there's the laser. It's a little slow down so that you can see this. Here's the laser, and he goes over, right? Gets no water, is pissed, right? Doesn't like it. Goes back. I mean, I cut out just a couple of these blanks so that you can see this, right? Laser, he goes over, right? I mean, it's really the most unbelievable behavioral control I have ever seen. Yes?
Do you think that the mouse is able to see the light coming on?
Question comes all the time, but of course, this is not the preparation, number one, you use normally, and number two, your control animals, of course, get everything and get the vector except the general rhodopsin, right? And that's how you control for these possibilities, right?
Have you ever blocked the acetylcholine receptors and see what's happening in these tests?
We get to this a little bit, right? Not while we are doing this recordings. You could not do this. Technically impossible, yes.
Have you reversed the nose poke?
Oh yes, I just mentioned this. Of course, that's an obvious question, yes. Reverse nose poke, stimulated on the other side. The other side doesn't do it. Reverse nose poke has no effect, right? So we were not having lateralization effects or something like this. Of course, very important question, yes. And there are many other controls when you look into the paper, to really show that this is specific, right? Because These trials where we stimulate are often only selected trials, either in blocks or selected because we want to see whether there is bleeding over of something to other trials, right? Things like this. So the number of controls is endless before you convince yourself you're not being, basically falling for something stupid, right?
So would this decrease the animal's association of the cue to the reward? Given that you're given...
They are so overtrained, they are working on this for 3 months before they get even implanted, right? So it's not an uninteresting question. If you were to do this chronically, you would shift the animal's behavior, certainly, right? But they're so overtrained. In a way, that's so automated, that part of the task now, right? That will be hard to show. Yes?
Did you only try to stimulate the right hemisphere, or did you also try to stimulate the left hemisphere to see if there's
Doesn't work. It is a right hemispheric thing. It really is, right? It really is. Okay, so we believe these transients are causal, right? They are deterministic. They make behavior, right? They force you to detect a cue, right? So this is a little bit, at least, beyond a classic neuromodulatory idea. And before we go ahead and talk about neuromodulation a lot, I suspect a lot of this also will apply to other systems. We just haven't managed to measure this yet.
Correct projections are increased by this stimulation.
No, we looked into this also, right? Because again, these are selected blocks or selected trials that are being stimulated. And so we looked, as I just said a second ago, whether you would have some bleeding over into the performance of non-stimulated trials. All good questions, right? It's all in the paper. Can we move on? I need more than 3 hours if we continue this way.
We had 53 questions so far.
Can you also measure the ECH
Neuromodulatory level?
after, so if you do this, can you get the next trial and see if there's really...
We always do. You mean if we are recording using the electrochemistry method? No, we can, no. That's absolutely impossible, technically, at this point. Absolutely impossible, right? Because the electrodes, because you're chasing such small currents, you can have nothing in the neighborhood that even generates electrostatic energy of any sort, for starters. And that's before you put in a laser and funny things like this, right? Okay, so let's move on. Let's move on. So, of course, we got really interested in how is it that such a small transient in one area can force such a big piece of behavior, right? In a way, that's something I had a hard time, wrapping my head around it, right? Why is one single transient, a pulse of acetylcholine, so powerful? And the way we started to address this question is by measuring field potentials via the same recording site we use for electrochemistry, right? And it actually was the Nicolelis group that showed us how to do this. And why are we interested in local field potentials? Because, as you will hear from other speakers, more than from me, because you can extract oscillatory activity from field potentials. And why this is important, I will tell you as we get to it, right? So that's what we did, okay? So the electrodes, we recorded electrochemical signals, and then we switched basically over to an EFIS and recorded in the same animal, in the same session, sometimes these local field potentials, right? And what we saw was really, really nice, right? When we have a cue that yields a hit versus a cue that is being missed, what we see is gamma activity that begins sort of in the middle of the cue and then bleeds into the interval to the time when the response ports come out, right? So this is really cool because gamma, of course, has been traditionally associated with decisional processes, right, since you asked this. And so the idea is that this gamma onset coincides with a post-orienting decision. Here was the cue, right, and you could say, okay, I don't want to do nothing with this cue, right, I say a miss, right? But if you decide, oh yes, this is the cue, it matches my memory, I should do something else now, that is a decision. And this post-orienting decision, we believe, has to do with this lasting gamma activity that's showing up in the middle of the cue, not seen in missed trials at all, right? Of course, we don't know whether this is cholinergically mediated. This could be dopamine, really, right, that makes this gamma. And so what we did, and this addresses another question that already came up, is that we infused either the M1 selective muscarinic, M1 selective antagonist olanzapine, or a non-selective nicotinic receptor antagonist, and found that it really is primarily and only the M1 antagonist that blocks the appearance of gamma in those trials. And also, so these infusions, once again, are in right prelimbic only, and these infusions alone were actually capable of decreasing the hit rates all just by themselves, right? From 82 to 65%, while mecamylamine did nothing, right? And so we believe, yes, that gamma is very likely cholinergically and muscarinically mediated, and we are making these gamma oscillations. Well, what are these oscillations really meaning? Well, I think of it in simple terms. In order to get the behavior done, you need a lot of neurons that start collaborating, right? Locally and across cortical areas. Of course, this is only locally recorded, right? So the cholinergic pulse allows populations of neurons in the prefrontal cortex now to say, okay, let's work together to push that guy through the detection process. And that's the power of this oscillation. That's why we are so interested in these oscillations. There's more to this. What we also found is called gamma-theta phase cross-frequency coupling. What does that mean, right? What that means is that in the middle of the cue, just when this gamma appears really clearly, it also begins to align, as you see here, the gamma is in blue, right? So the amplitude of gamma begins to align to the phase of the slower rhythm of theta, right? So now you have two oscillations, in other words, that are shaking hands. Why would that make sense? Well, think of it this way. If you go through a task, coming back to her question, where there's lots going on, right? I mean, there is a cue, you have sensation, you have decision making, right? You have all sorts of things. So all sorts of things need to be done by multiple populations of neurons that oscillate at different rates. But in order for them to work together, they have to shake hands. And the shaking hands is this cross-frequency coupling, right? And so we see not just gamma, but we see other frequencies, coming on board, shaking hands, saying, I work with you to get this detection thing done, right? So again, is this cholinergically mediated or not? So again, here you see gamma normally very nicely aligned to the phase of theta. Again, you put olanzapine into the prefrontal cortex, completely destroyed, right? The handshaking is completely gone. So the populations of neurons don't work together. Therefore, you're not making, a detection, really, right? So the idea is cholinergically generated gamma ushers a transition from orienting to the decision to generate a hit response, right? And I agree with you. I agree with her. This is only, part of beginning to see how multiple cognitive steps are really required for a detection process. But in rats, these are possibly the limits, and the cognitive machinery, should be better worked out really in humans, if you like. Okay, humans, right? Of course, you can take EEG in humans, doing the task, right? And look what we see, right? 200-300 milliseconds after the cue, just when you expect it, you see again that the amplitude of the P300, which is what's happening at this time, in the right side pops up, right? Just under the fMRI. Then you see activity also spilling over into the parietal area. And what you also can extract from this data, which is really cool, is that there's gamma coherence now between frontal and parietal area, right? So which tells you, in the rats we only looked at prefrontal gamma, but in the humans now we see that gamma in frontal and parietal start shaking hands, right? So the oscillation spread to other cortical regions in the attention system, just as you would want to see it in any task that has an attentional component. Cross-frequency coupling we haven't managed to extract from this data yet, but we are working on it. But this is another good example how you can use human data to, again, confirm that the ideas you deduce from animals are not just completely ludicrous, right? So this is cool, and I cannot believe that I have to speed up, but you guys are engaged, which is exactly what we want. It's fantastic, right? So neuromodulation. There is a neuromodulatory component, and we measure it with microdialysis. And when I say this, you will right away say, you just fool yourself. Because microdialysis has a lower temporal resolution. Just because you don't have the resolution, you now think, you know, this is for real, and maybe you're just fooling yourself and all you have are these fast transients. But we know this is not true, and I'll tell you why once I've explained this data. So what the animals are doing here in blue is the normal task, right? The normal sustained attention task. And this is the task block, right? And so when we measure microdialysis-based acetylcholine levels, we see a nice increase that remains pretty stable throughout task performance. And there's very little dropping in performance. And once the task is over, we go back, right? But say now you throw a visual distractor into the task. You make it really hard, meaning the house lights are flashing at 0.5 hertz, right? This is a tough task now, right? And we start the distractor in the second, we have it on in the second and third task block, right? What you see is now that the ACH levels are massively higher than in the absence of the distractor, right? First task block, no distractor, they're identical, obviously, right? But now we see the highest levels we ever see while the distractor is on. But while the distractor is on, the animal is not performing well, right? It makes a lot of misses because of the distractor. So you have less transients, right? So this cannot be the sum of transients because you have a smaller number of those right now. So this is something else and something separate. What time scale is it on? We really don't quite know, right? As Trevor said, we can now dialyze at 1 minute levels using mass spec for analysis. But we still don't quite know where it is, right? It could be somewhere in between tens of seconds and hundreds of seconds where this fluctuates. So what are these high levels of cholinergic neuromodulation do, really? What they do and what they are correlated with is how well you perform while the distractor is on and when the distractor is off again, right? So higher levels of cholinergic neuromodulation allow you to perform relatively better while the distractor is on, and more importantly, they allow you to go back to baseline once the distractor is off, right? So we interpret this in general terms as higher levels of neuromodulation mediate better top-down control, right? Task set maintenance, right? You keep your task online. You remain goal-directed. I really want to regain task performance. I want to earn water reward because I'm thirsty, right? All these things we mean when we talk about attentional control or top-down control, right? So your neuromodulatory levels, even though some of you start yawning, right, should be really, really very high now, right? Because you shouldn't think about dinner and tonight or whatever, but you should stay on task and say, I really should listen here. I made a trip, you know, from so far. If I'm not learning something here, you know, what's the point, okay? So this will be a theme now from here on out, that levels of cholinergic neuromodulation control, you know, the degree of top-down control, right? As you see here in animals, right? Is it true in humans? Well, here's the distractor task. What we see is Brodmann area 9 is activated specifically by the distractor relative to a flashing control in all of these things in humans. Again, there is a correlation with the distractor effect, and there is connectivity, which is interesting for the fMRI guys with the anterior cingulate cortex, because now you have a lot of conflict going on, right? And you have a lot of errors going on that you would not like to commit. And so it makes sense that the ACC is being brought on board to import prediction error computation and things like this, right? So it's just the same in humans, and again, it's all right hemispheric. So how do these two things talk to each other, right? We know quite a bit about this. So in blue, say you have neuromodulatory levels. Can be high, can be low, right? If they are low, and I'll show you data about this, poor attentional control, low detection rates, you will be very distractible, you have attentional lapses and fatigue. If your attentional control is high, neuromodulatory tone is high, good detection rates, resistant against distractors, I'll show you that too, task representation is good, and this is a new sort of theoretical perspective we have embraced. There's a very low propensity for doing something else, walking away from the task, right? As you stay here instead of walking out and go for lunch. Yes?
You said at the beginning that the sort of old view of acetylcholine is it's an arousal sort of neurotransmitter, and is, you know, the kind of thing that you're talking about here in terms of attention control. I mean, is that just not another way of calling that
I don't know what arousal is. If you want to use the word, be my guest, right? The specificity of the terminology here has to do with task performance. And let me show you some more data, you'll see, right? So the transience, to add this, and I should say, there's no relationship between the amplitudes of the transience and the neuromodulatory component because we cannot integrate the two measures, so we don't know how to harmonize the two ordinates, if you like. We just can't do it because it's from such different methods. But transients have good amplitudes and are more likely when neuromodulatory levels are high, right? And when you are, when you are not paying good attention, when your attentional control is low, then transients, they look bad or they don't pop up at all, right? So you're not detecting much as you would expect when there is low attentional control.
So I don't think I understand really well what is triggering this neuromodulatory level. Is the distractor that it triggers?
Yes, it's the distractor. It's things that make your life hard. If you're equipped to respond to things that make your life hard, right? Say everybody starts talking now while you still want to listen to me, right? You have to beef up your neuromodulatory levels in order to maintain and even increase your detection rates, in this case of what I'm saying, okay?
That means when it's more difficult, I perform better?
Yeah, well, if you don't beef up your neuromodulatory levels, and I'll show you data on this too, you'll be lost once the distractor gets on. We get to this. Let me show you some data. You'll get to this, even human data, right? So how do these things interact? We actually know quite a bit about this, and this also comes back quite a bit to what Trevor mentioned before. And the only unusual thing here is that from the little data we have, we think that it's the neuromodulatory component that influences, the making of transients. So acetylcholine neuromodulation modulates transient generation, right? So it's a two-step dance with the same neurotransmitter. There is, however, an intervening step, right? So the cues get imported into the cortex by this glutamatergic thalamic projections, right? The way things work, to the extent we can say, and we have quite a bit of data, is that the neuromodulatory component via a nicotinic receptor, a presynaptic receptor, and as Trevor said, sometimes they can be part of the neuromodulatory system. The neuromodulatory tone influences the degree to which, or the amplitudes of cue-evoked glutamatergic signals, which in turn then drive the cholinergic transients, right? How do we know this is true? Well, if you knock those out, this interaction is not happening, for example, right? If you knock out the thalamic input, you don't get transients, right? We have done all these things, right? So let me say this again in simple terms. You have your neuromodulatory tone, which should be high now, right? Which means you are, among other things, and Michael Hasselmo will possibly add some other things to this idea, you are now amplifying your thalamic inputs, my speech maybe, right? And therefore you are making transients, which means you're actually listening or detecting or perceiving the words I'm saying, so to speak, right? So it's a cholinergic interaction. The alpha-4 beta-2 nicotinic acetylcholine receptor is actually a perfect player for neuromodulation because it's not desensitizing. As a matter of fact, it's upregulating as it's being hit harder in the cortex, right? So it's really a great mediator, if you like, to influence the making of these transients. And it also is giving us, as you will see later on, a great idea about how to treat disorders that are associated with low attentional control, right? Which is through alpha-4 beta-2 agonists maybe, which can replace, basically a lousy cholinergic neuromodulatory component, so to speak, right? So that's how we think these two things interact. And from here on out, I will mostly talk about, the significance, translationally, of neuromodulation, but you will keep in your mind that the significance has to do with detection in the end, right? Because bad neuromodulation is bad detection of whatever your task is, and good modulation is good detection. So I'm skipping this. This just shows you what I told you before, that we have a glutamate event from a thalamic input, which, by the way, oh no, there's one important point I still have to cover. The glutamate signal is seen in all HIT trials, every time. But I already told you the cholinergic signal is only seen in those, unusual hit trials that follow these blanks in these incongruent hits. And the glutamate signal is also faster compared to the choline signal, right? So you can measure this also with electrochemistry. And again, if you're managing to, put different enzymes onto adjacent sites, you could measure them in one electrode, right? That would be hard, but it would be a fantastic thing to do. Okay, just to wrap up this transient business, I think the question you were walking yourself to before is this. Well, the brain already knows the signal is there, right? The thalamus has given the glutamate pulse. Every time there is a cue, you get a glutamate pulse. So why the heck do we need a cholinergic transient, right? You need the cholinergic transient to make these oscillations and to actually push the subject through the detection process. The thalamus alone is not allowed to do this. The thalamus just provides some post-sensory representation of the cue, right? But it's the cholinergic system that makes the decision for you, so to speak.
What happens on omission trials?
On mission trials?
Is there sometimes a transient rise and then a drop off when it's not followed by omission?
So this gives me the chance to say another thing with amperometry is that you can really, it's a little bit like electrophys in that respect, a noisy electrophys in that respect. You need to have a very precise event to which you are linking the transient in order to really extract it. So in an omission trial, nothing's happening, right? There might be an orientation to the cue. It's not giving you a transient, right? There's certainly no hit response. It's not giving you a transient. You might have a lot of noise, but it's not reproducible from trial to trial. So it's not informative, really.
So can you say that like noradrenergic system and the cholinergic system, acetylcholine has two phases of activity, tonic and phasic, and the shifting between two phases?
The tonic and phasic are loaded terms. Everybody means something. Let me just say this, means something else. Let's call them transient and neuromodulatory component, right? So they're interacting, so you're right, so go ahead. So the question is whether this exists in the noradrenergic system. Ask Gary about this, right? And I think it's a really, really good question, right?
I think the transient activation is like the phasic activation in other neurotransmitters.
Yeah, people mean something else with phasic, right? Because these transients are bound to behavioral events. If you want to call them phasic, fine. I would like to call them transients so that we are not confused with this, right? But you're asking the precisely interesting question, right? As I said early on, the reason why we don't know this about the noradrenergic or serotonergic system is because we don't know yet how to listen to the terminal. If we were able to, I would make a bet with you, you would find something like this, and you would find a deterministic component also.
Yes, Martin, this is a very interesting question, because also the dopamine inputs the cortex, the D1 receptor and the D2.
Yes.
I didn't explain this in detail, but these two are putting the cortex into two different modes. And it's analogous, although it may not be.
I agree.
D1 is kind of phasic, and D2 is kind of tonic, whatever you want to call it. How would those systems behave in the set?
We don't know, right? There could be interactions, and we have data which I will not discuss with you that show that they're moving in different directions depending on how the animal is dealing with cues, but that gets still a little complicated. So I suspect that there are local interactions, even potentially negative interactions, inverse interactions, but we don't really know, right? And the voltammetry guys owe us a lot of functional data on what you would like to call the phasic dopamine component, specifically in the cortex.
Based on this, it would be possible to check by the system you made and coupled in the same, in another structure, maybe with optogenetic, like channel reloxin, like isolating, for example, dopamine with channel reloxin, like with the ceramic and titanium structure.
Yeah, but you would have to know, in order to do this right, you would have to know how dopamine is behaving in real life, right? In that context, we really don't know, right? So it starts, you have to start recording and develop a hypothesis, right? Experiments that begin with, let's do it because we can, are always bad ideas, right? experiments that begin with, I have a hypothesis because I have recording data, and now I'm manipulating, that's better, right?
Already about the credit for that.
Let's move on, right? What determines whether a cholinergic transient is generated? Well, number one, the stronger the signal in salience, of course, the more likely you make a transient. And number two, and that's the point, higher levels of neuromodulation more likely make a transient, and a transient with bigger amplitude, right? So you pay attention, so to speak, in everyday language, and the stimulus is strong, and you make transients. Why are there no transients during consecutive hits? As I already mentioned, because this would bias performance very much to the detection side. It would take a little more time to explain this, but maybe I'm skipping this. This one is a headache for us, right? So the electrophys guys always want to kill us because we see these transients peak so late, right? I mean, when the levers come out and the reward comes down, we know that reward has nothing to do with it. We have tested this in 17 different ways, right? But certainly, there's no question that the peak is astonishingly late, right? Which is a little bit, again, even for the transient neuromodulator style. So what's going on here? Well, somebody even asked that question also, right? Yes, I think you did actually, right? What happens to the association, right, to the performance? So we suspect that actually once you did it right? So there's a cue, you hit it, you get reward. There's some feedback maybe, that's basically re-establishing that shift from monitoring to cue-oriented behavior, right? And maybe, but we really have no evidence, I'm just talking out of the blue, right? Maybe this late peak is really part of, a prediction error really computation. And there have been people in the literature that actually have been claiming that there is also a prediction error computation in the cholinergic system. But by now I'm believing that prediction errors are computed all over the brain, right? Because they're being found everywhere. Right? Because that's such an elementary thing for learning and behavioral plasticity. So why would we be shocked if it's also showing up in the cholinergic system? So these are just some of the questions I wanted to wrap up. So we're moving translational now. Wow, it's unbelievable. And again, the translational stuff focuses all on neuromodulation, right? Because that's what we can manipulate. That's where we have some clinical understanding. But again, you keep back on your mind that levels of neuromodulation influence, at the end of the day, and this is the thing, detection efficacy. Coaly Transporter, why would we care about the Coaly Transporter? Let's look at the synapse and how it works, right? So the Coaly Transporter sits in the membrane. There's tons of choline in the extracellular space, but it's the Coaly Transporter that controls how much choline is being taken up into the presynaptic site for making the neurotransmitter, right? So it's not the amount of choline that matters, it's the transporter capacity. And the transporter capacity is what matters more than anything else. How do we know? Because, we have had hemicolinium for many years. If you block the transporter, the cholinergic synapse is dead. And by the way, so will you, because you can't breathe anymore, right? Your parasympathetic nervous system is gone, right? Your heart will stop, all these funny things, right? So the Coaly Transporter is therefore of interest to us. What determines how well the Coaly Transporter works? Number one, it is, as we will see, the genetic variant you have in the membrane, right, for gobbling up choline. And number two, importantly, it's the trafficking of transporters from those intracellular domains where they mostly sit into the membrane, right? Because generally, as neurons have to work harder, like yours should do now, you are pushing more transporters into the membrane so that you can gobble up more choline to make more acetylcholine, so that you can now sustain higher levels of cholinergic neuromodulation, right? How do we know this is true? Years ago, we did an experiment where we had rats performing the task. Then we took out the brains and measured choline uptake, right, in many, many tissues. And wouldn't you know it, all we found is only in the right side we see in performing animals an increase in choline uptake over non-performing animals. And these were animals that were doing a pretty aggressive control test that was sort of in the middle. Nothing in the left, nothing in the striatum or elsewhere, right? So again, purely right hemispheric. And it was really very, very nicely formal. A certain increase in Coaly Transporter capacity predicted a 6% increase in hits to certain cues, right? I mean, really, really very cool. So, where do they came from? What surprised us is that when we look at the total amount of Coaly transporters in animals that perform the task and non-performing, that didn't change. So we knew that the neuron was not making new transporters, right? But something happens to the way transporters are being moved around. And so we developed a method to measure transporter in the plasma membrane and in those intracellular reservoirs, if you like, right? And one is called the LP1, the other one is called the LP2 fraction, right? And then when we look again at performing animals, and these are different animals, you see that only in the right medial prefrontal cortex you see an increase in the transporter density in the synaptic plasma membrane, right? So the animal says, I have to work this task, I need better neuromodulatory tone, so I'm pushing more transporter into the membrane, right? So it works, right, as advertised. And we have learned now in the last couple of years, working with a transporter specialist, Randy Blakely, a lot about how these transporters are being trafficked around. Some of it, of course, is depolarization dependent into the membrane, but there are other pathways into the membrane that are under control, talking about neuromodulator interactions by other neurotransmitter and signaling pathways, right? And that's still very poorly understood. But it is probably the case that specifically monoamines can influence transporter shuttling into the membrane, right? And recycling is through a classic Clathrin process, right? So why do we care about this stuff? And how do we really put some cognitive meat on this story? Well, there are humans, of course, that are regular humans that have 100% Coaly transporter capacity. But a guy named Okuda reported that in the human gene there is a SNP which, if you put it into a cell line, is giving you only 50% of your normal transporter capacity. And we thought there is no way, if you have that SNP, that you can compensate for this, right? You will never have the levels of neuromodulation a wild-type human have. And so we were predicting that these humans, as well as animals we made that mimic this deficit, as well as mice we now made that express that human gene, thanks to CRISPR, this has become so doable. We predict that all these guys that have this low capacity variance are super distractible, right? They would not show in humans the right frontal distractor-associated bold response. And I also want to later on talk a little bit, just a second, about the idea that if you have a bad cholinergic system, you're also more vulnerable for neuroinflammation. On the other side, so what happens if we make a transporter super mouse, right? Which we made by overexpressing the transporter, radically in mice. And more recently, we found a SNP in humans. I'll show you data about, performance data, we also think is an overcapacity SNP, right? So these humans have super transporter capacity, so we think they're resistant against distractors, stable performance over time, right? And cholinergic neuromodulation, we know at least from the mice, is very high, right? So is this true? I'm skipping this animal data because you have seen enough animal data. Let's do humans, right? It's way more fun, right? And we are already pressed in time, if you can believe it. So Cindy Lustig and I, 10 years ago, we started looking for these guys who have this I89V subcapacity variant. It's in 10% of the population, right? And we have now, I think, about 60 of these people, right? So it's just a classic SNP, right? Where you replace one nucleotide, which makes for another amino acid, which kicks the capacity down to 50%, right? So what's really cool is that these people, even before we genotype them, by the time we take the DNA, we let them complete a bunch of questionnaires, and they know that they are more distractible and have lower attentional control than wild types. They self-report more distractibility and more mind wandering in an inventory that has items like, I find it hard to concentrate when the TV is on or the radio is on, right? And we were very impressed by this, right? Every cohort we collected, again, self-reported this. And so we thought, okay, let's throw a real tough task at them where they really have to deal, like in those items, with massive natural distractors, really, right? And you have to do this task, so listen to me how this works, right? So here is a grid that changes direction every 800 milliseconds. But sometimes it's 1,070 milliseconds, right? So there's a little bit of a longer delay between two grid orientation shifts. And those are the ones you have to report, right? And while you do this, I'll show you some really, really exciting videos, you know, like this. Here's a long one, you saw it? Here's a long one, you saw it? Right? It's pretty tough stuff, right? For those people from the States, you will be enjoying the fact that we used dialogue from the Jersey Shore TV show also, which is a fantastic destructor for this, right? And so look at this data, right? This is non-destructor wild type, destructor wild type, right? They are dinged a little bit, but not too bad, right? These are these I89V folks, non-distractor. They're doing just as well in the absence of distractor than the wild type. But once a distractor is on, they're significantly impaired, right? And what is more, they also have relatively poor memory about the content of the videos, right? So they're distracted by the videos, they're looking at them all the time, but then they don't know what's in them, right? So this is really impressive. Okay, so we put him in the scanner, and amazingly enough, the right frontal activation that comes with the distractor is completely gone in these guys, right? They don't activate this area, right? So, of course, we would like to speculate that the failure to activate BA9 in these folks reflects the failure to activate the cholinergic neuromodulatory system, because they cannot, because they have such a lousy Coaly transporter. But of course, that's pure speculation. But the speculation is supported by mice that mimic this deficient transporter, and that they're incapable of increasing their neuromodulatory levels either, right? So this takes more work, but it's again a nice translation, at least the beginning of a nice translation. And we know now from my collaborator Randy Blakely that, not so surprisingly, this SNP is overrepresented in ADHD. And interestingly enough, and that bothered us for a while because we couldn't understand it, it's associated with greater depression severity. And we couldn't understand why this is for the longest time, because we thought this is all about attentional control. What does that have to do with depression? But I think we are having a little bit of an answer now, and I will get to this. But before I get to this, let's go for the superhumans now, right? So these are these humans that have this other SNP, which we think, for a number of reasons that would take time to discuss, is an overcapacity transporter, but we haven't really demonstrated this, and there's something difficult about this. But when you put them into the same video distractor task I showed you before, and these are real amazing data, they are absolutely unimpressed by the distractor, totally unimpressed. They perform at non-distractor levels, right? Literally, right? I mean, it's just a bar, but have I ever seen impressive data like this? I'm not even sure, right? And while they are doing the task, and so to speak, can ignore the distractor, wouldn't you know it, the memory about the content is better than that of the wild type about the distractor. I mean, it's totally amazing. And by the way, they also know it. They self-report that they are good at this kind of things.
Stupid question, but can you somehow control for the fact that some people might like Jersey Shore?
There are various distractors. They're not just doing one. That's a good point. No, we looked into this. We control for these things, yes.
So is there a caveat to this behavior? So basically, are they less flexible? Is there something that's going on?
Very good question. I almost had forgotten this. Benefits and costs, right? I mean, I think Trevor has been in the literature saying all along, if you turn the screw in one direction, you're going to pay for it, right? And the only thing we know about these guys is that their creativity scores are worse than the wild types. And the creativity scores of the low-capacity transporters are better. And it makes kind of sense, right? I think I discussed this with somebody yesterday already, right? If you have a low capacity distractor, you're not goal directed, you're always looking around, everything is so interesting, right? You're just a more creative person maybe than, us, tunnel visioned, high attentional control people who do one thing and around us the world goes under and we don't even get it, right?
I think I recall correctly from the paper that this is divergent creative discourse, right? Because convergent creative discourse.
Yeah, don't ask me, don't ask me. This was a student in this lab who did this, I know very little about this data, honestly, right?
In the highly distractible people, would increasing the overall cholinergic tone by giving a acetylcholinesterase inhibitor rescue?
The famous acetylcholinesterase inhibitor, right? And why are they not working, right? So let me just say it for once, because if you guys never forget this, the school will have been worth it, right? If you give an esterase inhibitor, you're doing a very crazy thing, right? You're increasing extracellular ACH levels tremendously, right? I mean, crazily, right? So what's this acetylcholine doing? The first thing it's doing, it's going to the presynaptic autoreceptor and blocks all presynaptic communication, right? Shuts it down completely. The second thing all this acetylcholine is doing, it's stimulating all muscarinic and all nicotinic receptors, non-physiologically extensively. How should that be good, right? The only thing I do not understand about the esterase inhibitors is why they are not doing more damage, right? But they're certainly not working. And we were just talking about this yesterday. And really, this is where, again, our translational research works. Because in our hands, and Trevor's I think is true, donepezil never really works, right, in animals, and it doesn't work in humans really very well either. You can squeeze out something at a certain dose, but it is not a good way to replace. Look, you heard me talking for 2 hours now, almost 1.5, about what she says calls phasic, what I call transient signaling and how important this is. And now you want to come and just, increase the extracellular levels like the Niagara Falls, get more water now, right? Cannot be a good idea, right? That's not how it works.
Martin,
yes.
Are they more intelligent? Do they have a high IQ performance?
No.
Which is interesting.
No. You would, of course, ask the question because you like separation of cognitive categories, and that's definitely happening here, right? Yes.
And do they have any impairments in other cognitive traits?
We know so little about them. Good question. Probably good question. This is all we have. Literally, yeah. Right.
What do the individual points actually look like in terms of, so at baseline level without a distractor?
Same as wild type controls.
But as in if somebody's worse at baseline, are they more distractible?
Don't know. Good question. Don't know.
Do you also see differences in their gamma power?
I have not done it. You guys have to come to the lab. We have jobs for you, right? Lots of them, right? I should see, right? Okay, let's move on just a little bit, right? Just again to show the mouse works too, right? Look at the overexpressor. So these guys have about 3 times more transporters in the membrane, and all the measures show they can do neuromodulation like super, right? You throw a distractor at them, second distractor block while the distractor is still on, they're right back to baseline, right? They are like these guys. They say, I don't care about your distractor. I can do this. And they're pumping acetylcholine like crazy, which I'm not showing you here, right? So it's really, again, translationally working, right? Okay. Why is this so funny? Okay. Stents, right. Stents. And so I have only one slide about neuroinflammation, but it's worth talking about it because the neuromodulators all do more than neuromodulation, right? And acetylcholine has traditionally, without anybody really knowing how it works, been associated with the inflammatory response, both in the brain and in the periphery. In the periphery, we know a little bit how it works. It has to do with spleen regulation and cytokine production, right? And so we thought, okay, if you are one of the 10% who have a low capacity transporter and your cholinergic system sucks, in other words, you should have a hard time dealing with brain injury because your neuroinflammation machinery should be abnormal, right? And so we thought, okay, let's look into this. And so we took mice that learned the task, and then they underwent 5 very mild impacts, right? On days that were just between 2 sessions, right? So one day they work the task, next day they get an impact, and next day they are back on task, and then they stay on task. And we made it so mild that the wild-type animals in black had no effect at all. There's a fly which wants to sit on my nose for some reason. Right, the Structor, right, right. And I don't have that good version. I know it, I'm a wild type, right? You got me, right? Exactly, right. Ah, man. So we dosed these 5 impacts that they had no effect on task performance in the wild type animals, but in those animals that are heterozygous in this case for the Coaly Transporter. So that's sort of a crude model of having a bad transporter. What you see is that every time they get hit, they get worse on the task. And in the end, they are here and they don't recover, right? And so we thought, okay, what's going on here, right? Why are they having such a hard time? And so I have a new colleague now, Natalie Tronson, who is an immuno expert. And so we started talking with her, of course, about cytokines, right? And cytokines, you know, are these disease signaling molecules in the periphery and the brain. So normally, if you're a wild type, and you bang your head, you will see some cytokine expression for a day or so, and then they go away, right? In these guys, this is 14 days after the last impact, and most of these cytokines are up and up and up, the red bars, right? So the brain of these guys continue to say, I'm sick, I'm sick, I'm sick, I'm sick, and they don't stop saying so, right? This is so exciting for us because we think that, of course, in all these many people that have brain injuries, right? I mean, military, sports guys, all of this, that the severity of the consequences have to do with the genotype of the Coaly transporters. Maybe, at least that's what something we want to look into this. And that's why I want to mention it to you, right? Because if you do stress research or even brain injury research, which will bring you sooner or later to the cholinergic system, I should have mentioned why are we interested in the cholinergic system when it comes to brain injury, because we all know that attentional impairments is one of the #1 things you measure even following mild traumatic brain injury, right? And that's the reason why everybody's looking at this. But now, right, so we bring this inflammation business into the story. And I'm also wondering, you know, if you are having this supercapacity version, which is more frequent, by the way, in 30% of the population, right? Are you a little bit protected against brain injuries? Is your inflammation system working better? We have no idea, but it's a fair question. Okay, and this is just for fun, really. So in humans, right, I can genotype you, but I would like to know what your capacity for cholinergic neuromodulation is, really, right? But I can't take synaptosomes from you to measure choline uptake, right? And so we were interested for a while to figure something out, what we can do in humans, because I want to do translational research. And so there has been reports that in T lymphocytes, there's a lot of transmitter-like machinery, right? Dopamine, you find acetylcholine there, and some other stuff, I forget, right? So they're a little bit like neurons, as a matter of fact, right? And so, wouldn't you know it, we looked into this, we found the neuronal Coaly transporter in these lymphocytes, and we developed a way to actually assay Coaly transport in these lymphocytes, right? So this shows the suppression of uptake by the Coaly transporter blocker hemicolinium, right? It's very nice. It's just like in synaptosomes. In fact, it's nicer than in synaptosomes, right? Is this really useful? Well, we only know this from animals. Though these are our overexpressors, right, which have more choline uptake in the brain, right, as you would expect, but they also have more choline uptake in their T cells, right? And so we are now all excited because we think we can actually, you know, in those humans that have these choline transporter genetic variants, we can actually measure the impact on choline transport in T cells, and maybe even find variants in the wild-type population, which we don't understand yet, and can relate this directly to people's performance, right? Which would be really super cool, right? So all of a sudden, I'm a blood cell laboratory, which is really no fun, right?
But you know, Martin, we did something similar with lymphocytes, D2 receptors,
Oh, yes.
and predicted performance. We sent it to the journals, no one believed us. But we think it's going to be our most cited paper in 200 years' time.
Yeah. This is interesting that you mentioned this, because getting this thing published was a nightmare.
Exactly.
Right? Because people will just not go for it. Right?
But it's true.
Yeah.
It's true.
Okay. Yes.
Is the flip side to this hampered immunity?
Flip side to
Flip
this?
side to having this different genotypes, which reduces inflammation but might also hamper immunity.
You're exactly right. But I only know it for the periphery. So the idea is if you have a lousy choline transporter, your T lymphocytes don't release much acetylcholine, or they cannot be activated, and therefore the spleen regulation of cytokine release, it's a whole different story, but this pathway is established, is very much impaired. That's exactly how we think about this. And the way we go after this is we are actually measuring choline transporter now in patients, in T cells from patients that have MS, multiple sclerosis, right? Because we think that's the best model to go after this. It's a very clever question. Okay, good. So this is why we're interested in the choline transporter. That's translation number one, right? It has to do with levels of cholinergic neuromodulation, and it has to do with the degree to which you can resist distractors or fall for them, right? And it may also have to do with neuroinflammation, who knows?
Is there any cholinesterase in this system?
I'm not sure I understand the question. When you assay those? Yes, I don't understand the question though. I understand the words, but I don't understand the question.
Destroying these acetylcholine.
You mean the T cell release? There are esterases all over your body, more or less selective for acetylcholine, right? Your blood is full with various esterases, right? So yes, the acetylcholine will be hydrolyzed in some form, right? Absolutely.
And the question was for the answer probably be no, but if you looked at microclean and exercise,
all good questions. I'm a bloody beginner when it comes to all this inflammation business, right? We will have to. I know this, right? Because Natalie, my colleague, is telling me about these things, yes.
Related to her question about esterase, I was wondering if anybody have looked at the esterase levels in these different genomes to see if they compensate for the transporter.
Fair question, nobody knows. Right? I was saying to somebody else the other day, one of the darkest holes, most unknowns we have in this business concerns esterase regulation, right? We just think it's there and it's powerful, but that cannot be the story, right? Totally. There's another PhD thesis or 10 in here, right? Okay. All right, let's do another translation where neuromodulation matters and attentional control matters, surprisingly, and this concerns falls in Parkinson's disease, right? So what you guys probably, many of you don't know, which I also had to learn, and learned by a coincident, totally unplanned discussion I had with a neurologist I know, is what the problem really is in the clinic now is to do something about the fall propensity in Parkinson's disease. This is a big deal. Because once these people fall, they break their hips, they go into a clinic, they go into nursery homes, it's very costly, right? L-DOPA does nothing to it, right? There's no treatment, nobody understands this. And in fact, the whole discussion started by a neurologist coming to me and saying, we have these falling patients, right? What do you think about this? And I knew a little bit about the literature in aging where it says the reason why older people fall is because they are having less attentional resources to now supervise their movements, right? And when you get older, I guarantee you will observe yourself that you will deploy more attention to control your movements, specifically if you walk around areas you haven't been walking around before, right? And so the idea is that the reason why these Parkinson patients fall a lot might have to do with the fact that in many of these patients, there are also cholinergic losses, right? That's how we all got started. Risk for falls in patients, which turns out to be important because the same is true for animals. Movement pauses, short freezing periods, slow movement speed, lower step frequency, poor posture, right? Less effective rebalancing after slips, and of course, attentional distractors and competing tasks. That's the classic paradigm how to make humans fall, specifically aged humans, right? So, you have seen this data before, right? These two guys with the lowest residual ACH levels are fallers, right? And they have the lowest attention scores, right? We have very little data about fallers because they don't want to come in, right? I mean, they are in our clinical trials, but they don't want to get out of the house, really, right? They are scared to hell, right? They have already fallen a couple of times, right? So, my colleague Nico Bonen, who is a PET scanner, he showed in 2009 already, and then nobody did anything about it, that falls regress to losses in the cholinergic system, but not in the dopamine system, right? And so that's how they came to me. Hey, you do cholinergic system, what does motor function have to do with the cholinergic system? And I say nothing, but it takes away the attentional compensation for poor movement control. Yes?
This is very interesting because for my knowledge, in striatum, acetylcholine is up in PD.
We get to this. Hang on for a little bit. We get to the striatum a little later, right? So the idea is this, right? You have, in these patients that are fallers, cholinergic losses. So your detection machinery, your neuromodulatory detection machinery is not good, right? And so exteroceptive and interoceptive movement cues are not getting well detected, not well processed, and not effectively transferred, therefore, to the striatum, right? In the striatum, they meet a network that also is deprived of dopamine, of course. These are PD patients. And what does the loss of dopamine do? Well, the easiest way for me to say, they become slow and have very low vigor gait, maybe some movement sequencing problems, etc. But together, this loss of cue information and low vigor gait, that results in impaired complex movement control and therefore in the risk for falls. So that's the simple way we now think about, so to speak, these interactions between losses in the cholinergic and in the dopamine system in these Parkinson's patients. And so we set out to model this in animals, right? And so we built this fancy task where you have beams that animals learn to run, like these rotating squared rods. Then we give them distractors, like this door frame distractor, which is actually something I learned the neurologists do in humans, because if you're a faller and you go to a door frame, they usually start freezing because they're distracted, and as they reinitiate movements, they make mistakes and have risk for fall. So we just modeled this, right? And I'll show you some of that, right? And initially, we did the simple thing, right? We just modeled the disease, which means we used the toxins to make relatively small dopamine losses in the dorsomedial striatum, in the prefrontal projection field of the striatum. And we also blew out about 50% of the basal forebrain cholinergic system. So the key group of animals are these DL dual animals that have, like the patients, basal forebrain, cholinergic and dopamine striatal losses. And we'll also talk a little bit about what the PPN loss in the pituitary-pontine cholinergic nucleus adds to this in a second, right? And so here are some data, right? So this is falls of animals running over this rotating rods, and I'll show you a video in a second, and have door frame distractors and what have you, right? So shams, dopamine losses alone doesn't do anything. Cholinergic losses alone doesn't do much, but again, the combination losses makes them fall a lot, right? And when we throw the animals also in the attention task, in those dual animals, the falls correlate with a poor performance, with a poor hit rate in the attention task, right? The poorer their attention, the more they fall, and the other way around. And we also know from histological analysis that if you have these dual lesions, we thought initially that it is the size of the cholinergic lesion that actually determines how much they fall. But it actually turns out that it's still the size of the dopamine lesion that determines this. And so the way we look at this is that when you have your cholinergic losses on top of it, you're now unmasking that your striatum is not functioning right. And you are now basically unmasking that you have lost your ability to throw attentional supervision or compensation at poor movement control, which is really cool. And just like in humans, why do they fall? They make more micropauses, slower traversal speed, less effective rebalancing, right? Just like in humans, right? It's really fantastic. I'll show you a video. This is a control guy, and this is where we are testing, how we're testing a secondary task there's a Fruit Loop on this platform, which, while he's running through this rotating rod, can pick up. And control guys, as you will see, do this just fine, right? He slips, but he rebalances, right? And goes on, right? Here's a guy with cholinergic dopaminergic lesions, right? He cannot do it, not ever. First things first, right? But they never learn to do this because they cannot rebalance, right? They cannot bring the paws back to the right position, etc., right? Which also, usually there is a neurologist in the room who at that point says to me, you guys do Parkinsonian falls in quadrupeds, right? Which are, quote by quote, inherently so much more stable than bipeds, right? And I just want to show you, if ever anybody says this to you, laugh him out of the room, right? Because yes, of course, right? A quadruped, if that's what it is, has a different topography, different kinetics. But again, just as in the humans, right? The rat model reproduces the risk factors for falls, right? And just as quadrupeds model so many other aspects of human behavior that don't look human in the first place, why would we not be able to model, the cognitive motor risk interactions that are responsible for falls? So don't be impressed when people throw this, reflexive argument at you, which I hear all the time now, right? By the way, the other question that comes up is, do they have sensory motor impairments? Here's the PASTA test, fitting for Italy, right? They have no problems with these things. We don't find any sensory motor deficits of any sort, right? I mean, their dopamine losses are fairly small, as you will see in the papers, right? So this is literally called the PASTA handling test, by the way, which was developed by, I'm blocked on his name, at University of Texas in Austin.
Timothy Shallett.
This man has memories, I tell you, it's amazing. Yes, Timothy Shallett. Thank you, Trevor. Okay. Yes?
Okay. So is the fall in the Parkinson's disease patients always associated with the distractions?
With?
Distractors? Well, how is it different from balance control?
Not always, but this is an effective way to bring it out, right? The other thing that will make a faller get into deep trouble is cued turning, not spontaneous turning, right? So if I'm a faller, I can walk, and if I want to, we'll just turn around and go, right? But if you say, whenever there is a tone, you turn, right? They will, beep, and they start freezing, and they don't know how to get started again, and they get into trouble with balance and rebalancing, right? So again, it's cued control of complex movement that matters, right? And secondary tasks bring this out very, very effectively, just like in aged people who are fallers, really, right? Okay, yes, there also have been some data, not very nice data, I will have to say, showing that there are losses in the cholinergic pedunculopontine nucleus, right? In animals that have complex movement deficits, right? In this case, these were MPTP-treated monkeys that on top of it got lesions of the PPN. So they're kind of dirty data, right? But we felt compelled and in fact pressured to look at whether PPN losses would actually add anything to the fall propensity we're already seeing in those dual rats. And we published this, and the simple answer is no, right? We're seeing effects of PPN on certain movement components, but we don't see an increased risk for falls, right? That would require a longer discussion why this is and whether, and that it needs to be followed up, but so far we haven't been lucky to bring the PPN really on board when it comes to falls, right? How do we treat this? Well, here we come back to that circuit, right? If they lost the cholinergic systems, part of it is, of course, you're losing the cholinergic neuromodulation that then translates into transient generation. And so since the key mediator is the alpha-4 beta-2 receptor, well, let's try an agonist, right, and replace the missing cholinergic tone. And we have done this with a compound from Abbott. Here we co-administered levodopa, but that's not even necessary, we learned later on. And with this kind of treatment, we are actually cutting down in animals the falls by about 50%, right? And this has been good enough for us to now go into patients, and we look into patients that have specific losses in the cholinergic systems, PET-based, and the only drug that's really on the market and we can use to test the idea is varenicline, the Pfizer compound, and we actually, have just started to run, my colleagues, that is Roger Albin and Bill Dower, they have started to run clinical trials to see whether we can reduce the propensity for falls in these patients. It's a true case of personalized medicine now, where you go after patients that have a defined low level of cholinergic neuromodulation, if you like, on top of their dopamine losses, right? So this is pretty exciting stuff. All right, striatum, right? So, of course, burning holes into a brain is always only a start, and it's a boring start, right? So we are asking the question, okay, if we have bad Q import into the striatum that interacts with poor dopamine levels, where does these things interact, right? And where they might interact is at the cholinergic interneurons in the striatum. Not because they're cholinergic again, but they happen to be, but because they're wired up to listen both to the cortex and, of course, influence dopamine levels and be influenced by dopamine levels, right? So this might be the element where the two twines meet, in other words. And what we began to do in order to study this is what so many of you guys do, is we're using TREAD to inhibit those interneurons and see whether this is good enough to actually reproduce faults in the model. And so I'll show you just a video that will give you the idea of how this data look like. And let's start with the video up there. So here's an animal, intact animal that simply runs this rotating rod. If it gets going, it did not, so let's do this again. Here we go. So it sits there, the walls come down, we don't want to touch this guy, they hate open fields, so they will walk to the other side where the walls are up, right? So this is an animal, intact animal, slips, but rebalances, recovers, right? Slips again, right? Slips again, it's pretty hard to see, slips again, right? Okay. So this is the same animal next day. Now we're giving the animal CNO, and again, there's the inhibitory M4 expressed by the striatal interneurons, right? Here he is. Slow falls, right? This is Aaron Kosinski's hand, right? Right, and here we go again, right? Falls, right? They just don't have good integration of time and movement, as you see, right? And so we now have data to show that all it takes is inhibiting the cholinergic interneurons in the medial-dorsal striatum, in the prefrontal projection field, and this will reproduce these falls, right? So that's the other translation we got so interested in. And that's the last one, right? And thankfully it is, because otherwise we would run out of time. And the last one is something you didn't see coming, I did not see coming. It concerns addiction, right? You're all dopamine fans, at least you were until now, right? And so you say, addiction is dopamine. What's acetylcholine having to do with this, right? And this is an interesting field for me. I got into a couple of years ago, thanks to all my famous addiction people around me. And the discussion always started with me being a little provocative and say to him, you dopamine guys, you talk reward all day long, you don't even know what that is, right? And you just think you explain anything by talking about addiction and reward. And by the way, you haven't been able to fix addiction in 20 years now, right? While you have been messing with the dopamine system. So something isn't right here, right? And I really got interested in this in part based on papers like this one from Trevor's group that are really fascinating studies because they show that even in healthy siblings of addicts, you find evidence for, in one word, poor attentional control as a trait. You also find evidence for corticostriatal abnormalities, right? And so my idea was always, coming from a cognitive neuroscience perspective, well, if you have poor attentional control as a trait, right, you will also have problems dealing with drug cues, right? Because drug cues, of course, over time in the addict, they become very salient in your perception. They become attractive and magnets, or as my addiction colleagues, Kent Barrage and Trevor Robinson would say, there's a lot of attribution of incentive salience to these cues, right? You really like them, right? But this also means you can't control them, right? If you are an addict and I'll show you a syringe, you cannot take your eyes off that syringe and your entire brain starts reverberating around, you know, maybe I should shoot up again, right? And pretty soon you get up, go out, find your dealer, and all of this, right? And on top of it, there is some evidence, including from an animal study we did many years ago, that shows that if you undergo extensive addictive drug use, your attention systems actually will further decline, right? And so my idea about addiction to a degree is that it's a vicious cycle of an attentional control deficit and drug seeking, right? You already have a poor attentional control as a trait, right? So you can't deal with cues. Your cue control capabilities get worse all the time because you're shooting up all the time. And so pretty soon, it is a problem of cue control, right? The way the people from the States will understand this, maybe you too, the others too, the way I always explain this to undergrads in a benign version, it's like beginning to think about the turkey Monday before Thanksgiving, right? You can't take your brain off it anymore, right? You think about the turkey every day until you sit down Thursday, and it's not even tasting well, right? But you're still shooting up, right? So make this pathological. This is the idea, my idea about addiction. And the other reason why I got that interested in this is because there is sort of an animal model for addiction vulnerability. And these are these sign trackers. The sign tracking approach has been developed largely by Shelley Flagel, who used to be a PhD student of Terry's, and Terry Robertson and Kent Barrage, right? So what are sign trackers and their counterpart, the goal trackers, right? It's very simple. You throw them into a Pavlovian approach screen, right? What happens is that the lever comes out, and I forgot, 30 seconds or so later, a pellet is being thrown into the hopper. But the cue is purely Pavlovian. You don't have to do anything with it, right? The pellet will come down anyway, right? And so the goal trackers, they are like the sane animals. They ignore the cue and go to the hopper, right? The sign trackers, they fall in love with that lever. They kiss it, they touch it, right? They embrace it if they could. So these are the animals, the sign trackers, that are particularly vulnerable, at least with respect to Pavlovian cues, for relapsing a previously acquired CS cocaine relationship and things like this. And the way my friends have interpreted this is purely dopaminergic and motivationally. They say, well, this is because you know, they have a dopamine-mediated, you know, falling in love, let's just say, with the cue, or this incentive salience attribution. There's a bit of an alternative, as I will show you in a second. And so because there are these elements, they are in all strains, by the way, so they're easy to screen with whatever rats you're using. And because they have sign tracking as an index for a psychological trait that has to do with this motivational process, we thought, okay, if these are addiction vulnerable animals, they should have some real attentional control issues, and maybe that's interesting, right? So we simply threw them into the attention task. Goal trackers, sign trackers have lower hit rates, and in sign trackers, the low hit rates correlate with a PCA index, which means the more extreme sign trackers they are, the worse they are on the attention task, right? That's cool, but what's even more cool is this, right? So this is, in red is a goal tracker, so this is, you know, the addiction-resistant animal, so to speak, and in green is sign tracker, and here is task performance over 60 minutes, and we split this into a good part, significantly, and basically a chance part. And what's important to see is that the sign trackers, they can do good, they just can't keep it up, right? And they fluctuate a lot, right? And here's again a good period for 3 minutes, and then they go back to being poor, right? While the goal trackers Just as you would expect for good attentional control, they do good and they stay good most of the session. Towards the end, they flip 3 times into a poor period, right? So these animals are really cool. I mean, they represent the clean cognitive performance component, if you like, of ADHD in a really great way. I'll skip that. You measure performance-associated ACH levels. Goal trackers pump a lot, sign trackers not during task, right? And look how small the error bars were, right? So usually the data we published were here, had big error bars, and we thought this is because microdialysis is such a lousy method. You get all those big error bars and everybody hates it. And now, of course, we know this is because we looked at undifferentiated populations. And who are we to think that one rat is like the next rat, right? I mean, I regret now that I came so late to sign and goal tracker screening because that cut all my variances by half, right? And that's something, I guess, that's important for all of us who are doing rodent and, of course, in a way, human research, right? So the idea is sign trackers have low cholinergic control, poor task set maintenance, and therefore the propensity for periodic random responding. So they are an animal model of low neuromodulatory cholinergic control. Yes?
What about the third of your animals that are neither sign trackers?
Yes, that's a very good question also. We call them the intermediates, and we know very little about them, and that's a real shame. Because there could be... You say exactly what, like, my colleague Terry Robinson says to me every second week, right? But nobody has ever time to look into these intermediates. Come to the lab, okay? So, somebody asked this before, right? So now, what do these low attentional control guys do with distractors? Nothing. They don't recover, meaning their post-distractor performance is not recovering at all, right? While the goal trackers, their post-distractor performance, you know, comes right back to where they were at the pre-distractor period, so they're really recovering. And the goal trackers pumping even more ACH when the distractor is on, while the sign trackers, of course, cannot pump ACH. And that's why they are not recovering, right? So it's the classic case of distractor vulnerability. Once you fell for it, you are done for, right? Because you don't have a neuromodulatory system that gets you out of trouble, so to speak, right? Okay, then we asked the question, why can't these sign trackers not use the cholinergic systems? And I thought for the longest time, well, it might be secondary to mesolimbic dopamine, because mesolimbic accompanies dopamine, you know, then transfers information to the basal forebrain. But since we are Coaly Transporter people, and we can look at Coaly Transporters, we thought, let's do it. And I have to say, we were really surprised by this. So what we did is, we took sign and goal trackers, we did electrical stimulation in vivo of the basal forebrain, so that we are activating the neurons and pushing transporter capacity up, right? And measuring this. And then, so after stimulation, we take the tissue and we measure Coaly transporter capacity, right? Here are the goal trackers in red, sham, and after stimulation, and you see that the Coaly transport goes up, right? So this is actually, you know, when you look at Vmax, you see that, you know, that's pretty darn good. I mean, they have, you know, 40% increase in capacity, as you would expect. This is really cool. The sign trackers, after stimulation, not get better, they actually have a tendency to get worse, right? So the cholinergic neuron doesn't respond the way it should, right? So normally, I stimulate your cholinergic neurons, you make your cholinergic uptake capacity going up. Sign trackers don't do this. So then we ask the question, okay, what's happening to the transporter during stimulation, right? So again, in goal trackers, you stimulate, and what's happening is what you expect. You find more transporters in the plasma membrane, right? Because the neuron has farmed out transporters into the membrane, so then the neuron can support the hard work. In sign trackers, that doesn't happen, right? What's more, and I hope you can follow this logic, when you look at the intracellular reservoir, in goal trackers, you lose them after stimulation on the inside. You should lose them, right? Because you farmed them out, right? Something has to give. But in sign trackers, you know, not only don't we get anybody on the outside being added to the outside, but we are also losing some on the inside. So they disappear from our assays. Where they disappear to, I don't know, right? They're not being captured by our fractionation strategies. But the fact of the matter is that sign trackers, and this is a hard neuromarker, have a dysfunctional choline transporter system. I swear you, this was never meant to be a choline transporter story. This only happened because we could do it, and we were, absolutely shocked to see this. Yes?
You made a very strong case it's maladaptive, and it's a disadvantage. But can I say, are there situations when sign tracking would be a behavioural advantage in an unstable environment, probabilistic shifting circumstances? Or sign tracking could be an advantage.
Just as we discussed when we talked about the humans with the low capacity transporter, right? If you're in the jungle and you have to watch out for lions, right, it's possibly a good idea to have low levels of attentional control as we define it. Absolutely, right? Maybe these sign trackers are also very creative animals, right? We just don't know it. I mean, you're absolutely right. There will be advantages, right? I mean, we are just, and I sometimes, think this is a bit of a problem. We were looking at everything with our modern values, sort of being put at high levels of attentional control. We think this is good and everything else is not so good, right? But in this case, the sign trackers are the addiction vulnerable animals, and that, in that context, this is interesting, right? Because we now think that low neuromodulatory cholinergic control is a risk factor, obviously, for addiction. And the dysfunctional transporter is the molecular machinery, to the extent we understand it, that underlies this, right? And so we thought, well, if it's so fundamental, why do we this complicated things and dialyze while animals perform the task? We should just reverse dialyze potassium to depolarize the terminals and should see that the sign trackers can follow. And indeed, that's the case. So here we start reverse dialyzing 100 millimolar potassium, right? So you see goal trackers, they pump out, and can keep it up, until pretty much the end of the potassium period. The sign trackers go up a little bit, then they fall right back down, right? So all you need to do is reverse dialyze potassium, and you see that your sign trackers have a lousy cholinergic neuromodulatory system, right? Does this has to do with the actual sign tracking behavior, right? And we were finally able to ask this question because the Vanderbilt group has made an inhibitor that actually one can use for pharmacological studies and given systemically. So it's sort of more of a negative modulator. In contrast to hemikalinium, you cannot administer. Animal will die right away because you're blocking the transporters. So when we take a bunch of rats and put them for 5 days to the screen, we have our usual split, almost 30-30-30, right? 10 sign trackers, 8 intermediates, and 6 goal trackers, right? Here's another group of animal, and we start giving them, prior to every screening test, this inhibitor of the Coaly Transporter, and now you see we end up with 19 sign trackers and only 2 goal trackers and 4 intermediates, right? So we can push their behavior towards sign tracking. What does that have to do again with attention and low attentional control? Well, think about it. You are the rat, right? You are doing the PCA screen. Lever comes out, food drops, right? Lever comes out, food drops. As a goal tracker, you say, lever? I don't care. I go for food, right? But the low attentional control guy sees this lever coming out, the only salient thing that's happening, throws itself at this lever and gets stuck caring about this cue. Eventually, there will be motivational processes, I suspect, that are superimposed over the saliency-induced attraction of the cue. But the beginning of being a sign tracker has to do with a low attentional control. Yes.
And on that note, I mean, if you have like an enriched environment, for example, right? Because as you said, in this case, the only salient thing that's happening is the looking out.
Yes.
So if there are other distractors, is there a chance that you would develop that kind of behavior?
Maybe. Likely, I would think. Nobody has tried this. Likely, yes, because there would be other well, it depends whether they're Pavlovian or whether, whether they are non-contingent and things like this, right? Because the Pavlovian contingency is absolutely critical. There's no question about this. So, for us, therefore, by the way, addiction becomes very much a cholinergic issue, right? And there's more data I have no time to show you. Which brings us back to the humans. If you are among the 10% who have that low, lousy choline transporter? Are you more vulnerable for addiction? Well, without knowing it, I would say, sure, that's a plausible hypothesis, right? And we are certainly interested in looking into this. We did something else, right? If you're a sign tracker, and if you have low and poor attentional control, you should have trouble with our Parkinsonian faults also, right? Because you don't have the attentional resources to throw at complex movement, right? So we tested this, and for example, if we have animals that learn to run a counterclockwise rotating rod, and then we give them for the first time clockwise, the goal trackers fall a lot the first time around, but then they learn it. The sign trackers don't, right? They don't have the attentional capacity to throw at this thing. And now for some entertainment, right? So we thought in order to bring this out more clearly in sign trackers, we need to beef up our attentional taxation of their movements. And so we developed this zigzag beam, right? And you got to see this to believe it. It's just complete fun, right? So, you know, he hates when the wall comes down, right? So this is the beam, stable, but you'll see, right? Makes a lot of slips, right? So this is one beam, obviously, for cameras, you see, right? Okay. So this is obviously a very different beam right now, right? Because now you have up and down rebalancing requirements. And now for the fun. Okay. So I can tell you sign tracker cannot do this. And the reason why they cannot do this, and we're still analyzing this data, is because the goal trackers, they actually halt their movement in order for the beam to get at the angle they like and the rotation point they like. So it's getting complicated, right? And so they are timing their movements over key elements of the zigzag beam. And of course, that is a very top-down thing to do, right? The sign trackers cannot do this, right? They're basically running through this thing. They never learn to time their movements through components of the rotating zigzag that are really hard to manage, right? And that is, for me, interesting because it not only brings timing now into our behavioral analysis, which is a very striatal thing to the extent we understand it, but it brings timing also to our top-down attentional control conceptualization, which I think where it belongs.
Can the high 89V mice do this?
We can't do it with mice. We have no machinery for mice, unfortunately, right? Good question, right? And that allows me again to say to the human folks, right? If you are having a low capacity choline transporter, are you at greater risk for complex movement deficits if you get Parkinson's disease or just if you get very old?
Have you genotyped these sign trackers to see if their cholinergic transporter genes are in?
We don't know what the genetics is of their choline transporter dysfunction. However, there is a guy at UC San Diego who got from us about 2,000 tails now, and who runs the mother of all GWAS studies to see whether we can pin this on something. It could be complicated because it could be any signaling molecule that controls the trafficking of these transporters. So that could be very hard. But it's a very good question once again, right? Okay, I'll skip this. Are there human sign trackers? Of course we are interested in this, right? And what we do is we bring the animal task to humans, right? So in the human task, this thing, which you can turn and rotate, pops out. It's like on some gaming machine, and you can play with it, and you get a coin here, right? It's just like in humans, right? And so what do we know about this, right? So colleagues of mine have tested kids, and you would not be surprised to hear that kids that are younger than 5 years old are all sign trackers, right? They touch this thing, love it, right? They know actually, in a way, that it's non-contingent, right? It's Pavlovian in our language, but they will be sign tracking. And as they get older, they are splitting up. We have done a little work with Synthilastic thinking about that we find an overrepresentation of sign trackers in smokers. That may well be the case, but there we have a little data, but this requires more work. And I have another colleague who is interested in people with eating disorders, right? And she actually thinks that she will have evidence to show that certain eating disorders, again, have an overrepresentation of sign trackers, right? So vulnerability for addictions of various kinds might be very much associated with sign tracking. And of course, once again, if you're a sign tracker, a human sign tracker, you might have a lousy transporter. You certainly have probably a low capacity for cholinergic neuromodulation. You're at risk maybe for various things, right? From addiction to movement problems, et cetera, right? So this is a very exciting thing in progress. So I come to my conclusions now, and also, again, want to talk a little bit about other systems, right? So what do we got? We got a deterministic component and a neuromodulatory component, right? We got pretty good translational models now, and things translate, right? I mean, you hear sometimes such criticism, for the red behavioral neuroscientists, and of course sometimes red behavioral neuroscientists do lousy, stupid work, right, to be quite honest. But we can do real translational research now if we are serious about this, right? So we have good models for high and low levels of attentional control, right? We have humans to double check on these models, right? We have definitely useful treatment approaches, right? I should remind myself and you that there were 2 of these compounds being tested in adult ADHD patients in phase 2 studies, and they worked both, right? Which again confirms precisely what we have predicted from animals. We certainly need to learn more about these oscillations, but they seem to be evoked by muscarinic mechanisms, postsynaptic muscarinic mechanisms, and we have very little pharmacology at this point about agonists or PAMs at this muscarinic receptors, and I'm sure Michael Hasselmo will tell you more about muscarinic receptors and the potential therapy that comes from that. The Coaly Transporter is a whole new target for pharmacology, right? Nobody's working on this, right? There are ideas you could have how to bring more transporters into the synaptic plasma membrane, and that could be a very good thing for various disorders. And of course, right, we know the striatal interneurons are the critical for everything, right? So why are we not looking at efforts to actually beef up the output of striatal interneurons, at least and specifically for these Parkinson patients? And so back to other systems, right? And you have seen figures like this in Trevor's introductory talk, right? There are parallels that tell me things will be a bit the same with the other systems, right? Small number of neurons, right? 5-HT, dopamine, right? As Trevor said, noradrenaline, right? Hugely collateralized systems. The tradition says, well, they do all the same, and what they do we find out by recording from the cell bodies, but maybe not, right? Maybe what they really do we only find out once we can listen to the terminals, because maybe the noradrenergic system or serotonergic system doesn't do the same thing in all areas, but it's the local cortical circuitry that tells the noradrenergic system what to do and when and how and what for, right? Surely the conclusions, if you are ever able to listen to the release of these neuromodulators in real time, will not be, at least unlikely be super radically different from some of the ideas, Trevor already explained to you, but who knows, right? What we really need is a method to listen to the terminal, right? And to understand, what we also need is to better understand how the terminals of these huge ascending, hugely collateralized systems are being integrated into cortical circuitry, and we don't know this at all, right? I just don't think by recording from a small number of neurons and ignoring the other side of the phone line that's also wired up, we will really ever understand how these systems work. And as Trevor also said, all these systems or deficits in the systems are increasingly associated with fairly specific clinical, cognitive, and affective symptoms, right? Which isn't easily translated back into a simple, functional hypothesis about the entire neuromodulator system, about the entire serotonin or noradrenergic system, right? And so much of this specificity, I just think will have to come from being able to listen to the terminal. And lastly, and some of you guys said this last night, and I was very happy to hear this, and Trevor mentioned this, of course, we are all sinners by being neuromodulator isolationists, right? But when we look at the anatomy, even roughly, all the systems get, of course, as Trevor also mentioned, get top-down input from prefrontal cortex. They're all connected, right? The basal forebrain system gets input from all the other nuclei, though all these neuromodulator systems work in orchestrations, right? And yes, we put out the point, and everybody accepts it, but we are all so far, because it's hard enough what we do, but we are all sinners by not looking at the systems in orchestration. And that's something I think we really will have to change in order to better understand how neuromodulator systems work. And some of you guys said last night, I like them all, or they interact anyway, so why would we have a preference for one? And you guys win the prize for last night, right? Because you're exactly right. Okay, so neuromodulator systems mediate multiple functions based on neurotransmission on multiple time scales. Cholinergic systems, we have 2 time scales. Are there more? Who knows? Maybe, right? I mean, we are only scratching the surface. Regional and temporal selectivity of neuromodulator functions are largely due, I believe, to local target circuitry and the presence of heteroreceptors. Again, something that outside the cholinergic system remains very poorly studied. Plus, again, coming back to Trevor, explosion of receptor subtypes, right? If you count alternative splicing, the number of serotonin receptors is, what, in the 30s, Trevor, or something like this, right? So there must be a differential style of communication we don't understand yet at all, and we certainly have not captured with any of our current hypotheses. Challenges and opportunities for drug treatment, right? If we are missing entire timescales of neurotransmission and the receptor machinery that comes with it, we are missing entire ideas how to treat patients. And again, as I just made this point, interactions among neuromodulator systems, that's just really a real, poorly investigated field, and we all have to go there. I just want to show you some of the folks in the lab. Vinny, who's now in Temple, he started all this electrochemistry in my lab. Giovanna, who is not so far from here, used to be one of the early sign tracker dialysis folks. This is my Coaly Transporter man. This is my dialysis HPLC-MS specialist. These 2 guys worked on the oscillations and on the transience a lot. This is my Parkinson man. You saw his hand. And used to be a student of Cindy Lustig, who is now at UC Berkeley. She worked on those Parkinson attention data, and there's still another paper to come. This is the neurologist, in addition to Bill Dower, who does the clinical part of our fall Parkinson research. Randy is my transporter colleague. He's a molecular biologist who is totally essential to our interest in molecular transporter biology. Cindy is my faculty colleague. She's an fMRI cognitive neuroscience specialist. There would be no human research in my lab without her collaboration. And Terry is an addiction guy who I slowly turned into a cholinergic, attentional control guy. And I thank you so much for your attention.